Good afternoon, everyone, and welcome to OXP's Capital Market event. I'm Aurélie Charpentier, Head of Marketing and Communication at OXP, and I'm delighted to be here with you today. For everyone joining us here, thank you for coming, despite the tube strike, and to our webcast audience joining from around the world, thank you for being here. Today, you're going to hear from OXP's senior leadership team and industry-leading voices. Let me take you through the agenda. We'll have 6 presentations broken in 2 sessions with a 15-minute break in between. First, you'll hear from our Chief Executive Officer, Dr. Frank Matthias, who will provide an overview of where OXP is at today and our market positioning. Second, Professor Luc Vandenberg from Harvard Medical School will join us online. He'll provide an independent perspective on the current advances in cell and gene therapy and the opportunities ahead. Third, our Chief Innovation Officer, Dr. Kyriakos Mitrofanos, will show you how innovation can enable client delivery. You'll hear about next-generation platforms, AI, automation, and new markets. And fourth, our Head of Process Development, Dr. Nick Clarkson, will demonstrate how process development excellence and a client-centric approach can deliver value. We'll then have our first Q&A session of 15 minutes with these 4 speakers, followed by a 15-minute break. After the break, you'll hear from our Chief Business Officer, Dr. Sébastien Ribaud, who will take you through a deep dive into the cell and gene market opportunity, our business pipeline, our conversion rate, and why clients choose OXP. Sébastien will then sit down with Arun Das for a fireside chat. Arun Das is the Chief Business Officer at Cabaletta Bio, one of our key clients. Sébastien and Arun will discuss our partnerships, why Cabaletta chose OXP, and the value we'll deliver. And finally, our Chief Financial Officer, Dr. Lucy Crabtree, will cover our strong financials and attractive outlook. We'll move to our second Q&A session, 30 minutes, with Frank, Sébastien, and Lucy. Frank will then return to summarize and close the session before a drinks reception. During the Q&A session, we'll be taking questions from the room. For our webcast audience, you can submit your questions throughout the day, and we'll read as many as we can today, for we will answer to individuals after the session if we couldn't get to their question. And with that, I will now hand over to Frank to open the session.
Thank you so much, Aurélie, and hello, everyone. Nice to see you here despite the strike. It took me 2.5 hours this morning to come from the airport, but I'm on time and I'm happy about that. So it's a pleasure to welcome you all to our capital market event, and we really appreciate that you took the time to come to follow this session. So we have prepared a very nice agenda, as Aurélie just showed you, and we will share different perspectives on the company. So first, what are the market opportunities we have and we see ahead of us? Then we will discuss the progress we have made over the last few years in building a pure-play CDMO company, and then we will also try to show you why we are so confident in delivering long-term sustainable growth. But before we start, and Aurélie has already mentioned it, I want also to express my gratitude to our guest, to Luc Vandenberg, who will join us virtually. He's from our Innovation and Technology Excellence Board, to kind of advisory board to the company, and of course to Arun Das. You presented him already, Aurélie, Chief Business Officer at Cabaletta. Thank you for taking the long journey to visit us also today, Das. Arun, thank you so much. So let's start, and because some of you might not be, you all know this one. Yeah, so please consider it. Yeah, thank you. So perhaps some of you are not so familiar with what we do and what the cell and gene therapy space is about. We thought to present you a short slide to make it evident what kind of role, crucial role to say, we play. So just in one sentence, cell and gene therapy is a medical approach that treats diseases by modifying, replacing or introducing cells and genetic material to restore a normal function or help the body to fight illness. That's what cell and gene therapy is about. And what you see here is that these therapies are about fixing diseases at their biological source. So instead of just treating the symptom, you try to correct, to replace, or to add what's working not so well into the body. And this has the potential to cure. I will come back to that because I believe this is an important part to understand. So we play obviously here a central role, as you see, by providing the viral vector, which you can consider to be a kind of vehicle to bring the code, the genetic material into the body. That's what we do. In the middle, we just focus on the development and production of viral vectors. So let's have a short look at the market we are in currently. And I believe it's important to mention here that what we are seeing across the cell and gene therapy market is not just a short-term cycle. It's everything but not that. It's really a sustained expansion of therapeutic pipelines across all stages of development, from preclinical programs up to commercialization. So the number of products is estimated of programs to say it's estimated to grow, and you see that on the left-hand side here, 9% year on year over the next 5 years. So by doing that, they will fuel demand for high-quality manufacturing partners. What we see also in the middle is, at the bottom of the middle, is that currently 38 cell and gene therapy products are already approved globally. And what we see also is that we expect a high level of development of the revenues for the outsource. Here is on the right-hand side, it's just the outsource potential market growth, which is supposed to grow. And whatever source you take of data, they are very similar in what they say, from 4 to 10 billion in the next 5 to 6 years. So this will be 18% growth on average every year. So what we see also, it's a lot of dynamics in this market. Cell and gene therapy M&A deals just in 2025 up to 12 billion in value shows that there's a lot of dynamic in this market. Now, coming to what OXB is about, and I think it's important to remember that our vision is to transform lives through cell and gene therapy. How? By helping and by enabling our clients to deliver life-changing therapies to patients. So this field is fundamentally shifting the paradigm of medicine, moving beyond just symptom treatment towards treatment with potential to address the disease at its source and to cure. We, as a company, provide then the expertise, quality, and manufacturing excellence needed to translate this scientific innovation into a scalable commercial reality. So we have done that for more than 30 years now. We have been leading innovators in viral vector design, process optimization, and large-scale manufacturing. I'm sure Thierry and Unique will say a few things about that. It's part of our long-time history. And today we focus on best-in-class capabilities, scalable platforms, and state-of-the-art facilities. And our strategy is also very clear. We want to lead the cell and gene therapy CDMO field as the trusted partner through an unmatched quality and innovation. So our focus is also to build long-term partnerships with leading innovators. I want to mention you, Arun, here with Cabaletta Bio, because I believe it's a strong example of what a good partnership can bring for the benefit of, at the end, the patients. So if we go to a little bit of history, because I believe this history is also important to understand. As you might know, everything started in Oxford in 1995. It was a spin out of the Oxford University. And over the years, out from this origins at the end, from the spin out, we went through a lot of progress. We achieved a lot of milestones in cell and gene therapy, and we have been consistently evolving alongside the industry itself. I believe that the most important transformation was somewhere between 20 and 22, when the board of the company decided we need to take a decision. Do we continue to do product development for our own, or are we going for CDMO business? We decide to become a pure play CDMO company, and this brings us in the unique position in the field. So this has, of course, required strategic clarity and execution. Building over 30 years of expertise, we have continued to strengthen our global CDMO network and manufacturing footprint through strategic investment and targeted acquisition, as you know, in the US and in France. I will come back to that. So by doing so, we have broadened our technical capabilities across vector platforms. We are today vector agnostic. We can do every kind of vector, and we have built an integrated international network. And at the same time, and our CFO will certainly report on that, we were able to deliver 33% growth in full year 2025. But if we look back to 2023, we more or less even doubled our revenues within 2 years. So I believe I can say today that OXB is stronger, more focused, more scalable, more globally integrated than never before. And as such, we are extremely well positioned to continue to capture substantial market opportunities ahead of us. Just a few words on our global site network. As you might know, we have now facilities in UK, in France, and in US, and I wanted to show you how this came together. So everything started, as I said before, in Oxford in 1995. Today, Oxford will be our center of excellence for lentiviral vector development and also production up to commercialization. We are also able to do other formats, but it's a center of excellence here in UK. Then through the acquisition we did in 2022 of the homology CMC part, we acquired a center of excellence, or we acquired a site in US, which we now developed to center of excellence for AAV. I believe this was an important move because you cannot be considered as a global company if you don't have a footprint in US. At the same time, and there's a little bit due to the Brexit, we also decided to make an acquisition from Institut Mérieux in France by acquiring 2 different sites, one in Lyon and one in Strasbourg. And you see that the vector focus here is AAV and lentiviral vector. We can do both, but we can also do other like oncolytic and MVAs in Strasbourg. And finally, and I'm sure this was a game changer, we made this acquisition last year at the end of 2025 of a site, state-of-the-art site in Durham, North Carolina, which we acquired from Resilience. And I said it already, I'm sure this was a game changer because it has increased our awareness immediately in the market. We need to know that this is a fully FDA-approved commercial scale facility already with additional fill and finish capacity. So this allows us to really bring end-to-end offering in the United States now. And the way we see both, Bedford will be the center of excellence for AAV development up to GMP. And once it's in the GMP status, it will be transferred to Durham for production. That's the way we see it. So we are now globally in the western part at least of the world, and we are able, with a high level of flexibility, to serve all our clients, whatever the level of their clinical development is, or if the needs are for commercial or for development, we are able to do it from all the different geographies. And as always, behind everything which is successful, you have people. And I have to say that here, as you can see, we have a highly experienced management team. We have highly experienced people, around the different geographies. And I have to say that I'm extremely proud to be working with such inspiring people around me and us. And you will hear from some of them later from the team about what their experience are in financial markets, viral vector development, viral vector manufacturing, and the broader CDMO space. And this brings me to my last slide, a little bit as an introduction to the different presentation you will see alongside the afternoon. I believe it's important to see that our performance, the one which we consider to be an outperforming performance, is coming from 4 different pillars. They are mentioned here. So first is our global scalable infrastructure. This is the foundation that enables us to support programs from early development up to commercialization or up to commercial supply. Second, we have process development excellence, and we will show you a lot of data in this direction, and our ability to solve even very complex technical challenges. We have the third block is about our innovation-led platform, which ensure that we continue to evolve and stay ahead of what the client needs, but also of what the science is doing. And fourth, our commercial momentum, which reflects the strength of our pipeline and of our order book. And I'm sure that Sébastien will tell us a lot about that. So these are the 4 blocks, the 4 areas we really defining how we operate and where we focus as a business. And having said that, I would like now to introduce our first speaker today, a guest speaker in this case. So I hope that Professor Luc Vanderberg is online. He's associate professor at Harvard Medical School and an associate member of the Broad Institute of Harvard and MIT in Boston. Professor Vanderberg is a leading expert in the evolution of cell and gene therapy, and his lab work is focused on improving viral vector design and use. We are very happy to have him being part of our scientific advisory board. He's joining us today in a live call to discuss with us cell and gene therapies, how they are maturating and to impact a growing number of patients. And those roles CDMOs have to play in fulfilling the potential of this modality. So, Luc, the floor is yours. I would say in this case, the screen is yours. Yeah. Thank you so much.
Thank you, Frank, for that introduction. And it's a great pleasure to be here at this event in Oxby. I'm sorry I can't be there in London with you. We can switch over to the first slide. I was asked to provide an external perspective on the state of the art in cell and gene therapy by Oxby. And of course, that field of cell and gene therapy is the field that Oxby is providing essential services in, as you've just heard. At the same time, beyond from this external perspective, I am, if you will, a prototype end user of the services that Oxby provides. I often seek out manufacturing services from companies like Oxby and the likes. In terms of the title here, maybe if you go back to the title slide, please. Gene therapy is at an inflection point. And as is usual during periods of transition, this leads to a lack of clarity in terms of where the future trajectory leads us. It's my goal here today to share with you where my compass points and provide you then also with some data points that informs that opinion. So on the next slide, I'll give you a brief introduction as to where I'm coming from. I'm an academic primarily with training in biochemical engineering and virology, and I live in the field of molecular medicine. Originally from Belgium, spent most of my career on the East Coast in the US, where I eventually became the founding director of a center called the Grousbeck Gene Therapy Center at Mass General Brigham in Boston, where I'm also faculty at Harvard. I'm an engineer by training, so most of our work really is around building technologies, designing these technologies. We study these technologies as well. But importantly for this discussion, we try to translate these products. That last word there on the slide on the right. This process translation is building science out into drugs, into the clinic. So on the next slide, I described to you where this journey has taken me over the past 20 years on the industry side. I just talked about kind of my academic paths, but I've been fortunate to see several of these technologies and experimental drugs progress through development, and in one case, even the marketplace. Sometimes this happens through partnerships, like is the case with this vector kind of in the middle there, AAV9, that was incorporated into a drug, Zolgensma, which Novartis sells. And sometimes that path leads us to the creation of a new company, right next to it on the right, Akuos, a hearing gene therapy company, which is leading one of our technologies through the clinic, and that I still remain involved with even after its acquisition by Lilly. So on that journey of each of these programs that are listed here, a critical step is solving for your manufacturing needs. And that is a critical component all the way from the laboratory down to eventual market authorization and beyond in terms of commercial supply. On the next slide, let us maybe try to get to the same page real briefly here by providing you some of the key concepts in the fields. And I'll try to keep it relatively clear. I don't think we need full depth to have a sufficient understanding here in terms of what gene therapy is. The goal of gene therapy is really no different than any other field of drug development. We develop drugs for diseases where there's a need to develop these drugs. But unlike other fields in drug development, we have an active compound in that drug that is not a small molecule. It's not a protein. And of the traditional drugs that you're familiar with, it is a gene or it is a gene-modified cell there in the middle on the left. Why is this compelling? Why do we need a different modality than the drugs that we have? Well, cell and gene therapy has the potential to address, as I think Frank also referred to, the root cause of disease. It allows us to go after diseases that currently there are no treatments because they're technically hard to get to. And another aspect that I think speaks to the imagination, it has the potential to be durable. A one-time intervention with a lifelong benefit to the patients. There we say, and this is a word we use very carefully, in the best case scenario, a cure. So next slide, I will highlight to you why this hasn't been done before. Well, the challenge is threefold. It's called delivery, delivery, delivery. It's basically how do we get a gene safely and efficiently into the body to have that therapeutic effect that I just talked about. On the next slide, I'll share with you the kind of the solution that the field has come up with. And that's summarized here. The solution really is called a vector, illustrated on the top right there. A vector is a vehicle that facilitates this delivery issue. It's a post-van, if you will, that shuttles in a gene to the cell. The blueprint of a vector looks like this there on the top right. It's got an outer shell, which is the vector, and it protects and it guides the internal therapeutic cargo, illustrated there in orange in the middle, and that's the gene. This vector is either applied directly to the patient in a field called in vivo gene therapy, as you can see there with the arrow circling down to the right, to the left. And on the right, there's an intermediate step called ex vivo gene therapy, where that vector is applied to cells outside of the body, and those cells are then delivered to the patient in this field called ex vivo gene therapy. Now, OXB holds the expertise and the capabilities of producing these vectors at a purity for use in humans, and at a scale to support products through testing phases and eventually into commercial. The most common vectors in use are viral vectors, and these are inspired on naturally occurring viruses, but they were neutered to eliminate the noxious features of the virus. And two types you will hear throughout the presentation. You've already heard about AAV and lentiviral vectors are the most common ones, but OXB provides services in a variety of other ranges. So on the next slides, I'd like to give you just a little bit of the human elements of how gene therapy, cell and gene therapy can impact lives and is already doing so on a growing scale. I won't do it in terms of the numbers that Frank presented, but I'll do it in terms of these vignettes. Starting on the top right, this woman, Emily Whitehead, you can look her up, as one of the first patients treated with a CAR T cell when she was very young and suffering from an otherwise fatal pediatric cancer. Now we have several CAR T cell therapies, as you know, available globally, and they are making a market difference for these patients. Moving to the middle, spinal muscular atrophy, a fatal genetic disorder that leads to death, often in the first few years of life. A single administration brings therapy to these patients. Now, on the flanks and on the bottom here, two examples from sensory disorders. All the way on the bottom right, and we'll come back to that later in the presentation, hearing. Recently, there's been an approval just about a month ago from Regeneron in this field. We have been active in this field as well. On the left here, a particular story from just a few weeks ago. This is a drug that's now one of the first gene therapies ever to be approved, Luxturna. I had the privilege of teaming up with my old mentor, Jean Bennett, the woman who developed this drug and eventually brought it to a company, to Roche and Novartis, to commercialize it. Here we were reunited with one of the first patients who received the drug in both of his eyes when he was 9 years old. I was a translator and helped during the surgeries. And now I met him when he was 26 years old at a celebration event for Jean and Al, husband and wife who developed this, in really a remarkable story. This was not a cure. You can see the cane there that he's holding, but he gave testimony, Yannick, his name is, of how this changed his life to lead an independent, productive life. He's a psychologist now. He went through a career path, found a partner, and is now living independently and working in a professional context, something that would not have been possible without this drug that gave him back his sight. As a small vignette, just 2 weeks after this event, now a month ago, Jean and Al, together with the team of Luxturna, received what's called the Oscars of Science. This is the Breakthrough Prize in Sciences given out in California by the Sergey Brins and the Zuckerbergs, a $3 million award. In any case, in the next slides, let's go back to the science here. And this is just a brief iteration of the 50-year-old history of the field. While this is an innovative and ambitious field, it went through various stages since the 70s. Foundations that were laid, first steps that were taken, some stumbles in the early 2000s, and now, and this is going back to the title of the talk, a pivoting from a stage of maturity. We have drugs that work, we have technologies that work to some extent, but we're really now transitioning into this industrialization phase. And that is one of the pieces of that inflection point that I described in my time. So let's go into the inflection point and what's behind it in the next slide. It's indeed one where we now have all of these things that have happened, overall quite good things, validation of the science behind this ambitious field, annual markets north of $1 billion of certain drugs, dramatic clinical outcomes, increasing investment and acquisitions in areas like CAR-T, neuromuscular, and ophthalmology. But we also see clear signs of hesitation, commercial hesitation, and these have to do with safety events, these have to do with pricing, these have to do with market access. And these are legitimate, I would say. These are concerns that are legitimate. I'm not here to kind of gloss over those and tell you that these are overreactions. No, there is a real reason behind this moderation. However, I would be remiss to not highlight the clear opportunity that's there as well. And particularly that is sometimes overlooked in this window of time. The opportunity stems from the fact that the investments of an era of exuberance, and this is probably an era somewhere from the 2015s to a few years ago, where massive amount of investments have led to scientific and commercial hurdles that have been dramatically overcome, and that now the field is really primed to have an expanded impact in terms of biopharma. So in the next slide, let me walk you through some data points behind this. The first generation of cell and gene therapies has identified several limitations in terms of delivery, the types of cargo we can deliver, and how we bring those to patients. But as I mentioned, a lot of these areas have been significantly matured because of the technologies that have been invested in. So in the next slide, I'll give you a few short vignettes, and I'll briefly guide you through the scientific slides. This is actually some of our own data, and I understand not all of you are scientists. It's my full intent to make you a scientist by the end of this talk, so bear with me. But let me point you to actually the scientific slides, the histology on the right there. There you see two columns, liver and muscle, and you see 3 generations affected. I'll briefly walk you through it. When there's more brown there, it means that the targeting was effective. When there's little brown, it means that it was not effective. I tell you now that muscle drugs that are emerging today and coming on the markets have been based on this first generation vector that you see there. Barely actually reaches the muscle, largely gets trapped in the liver. That investment that I talked about before has led to several parties developing a second generation vector that at least gets you in the muscle significantly better. And then a third generation that shows that you can actually delete the liver where it doesn't even belong when you develop a muscle drug. So we have an uncanny ability to target these innovative medicine. And what it does is it effectively overcomes some of the main issues. 2 issues are that because of this limited capability of that first generation, we have to dose massive doses to the patients. That leads to high cost of goods and toxicity. And guess where? That toxicity shows up in the liver. So you can imagine the impact that a third generation vector could have for diseases like Duchenne muscular dystrophy. Next slide, just very briefly, a similar effort is ongoing in terms of the cargo, what goes on. So not the vector on the outside, that outer shell, but also what goes inside. Up until a few years ago, we could really only do this gene augmentation approach. But I'm sure you've heard of things like CRISPR and so forth. We now have abilities like gene editing, RNA-based approaches, sophisticated ways to control genetically where our target goes. And that is really because of a revolution in molecular medicine. In the next slides, I want to make the point that the opportunity goes beyond the science. Because of the appeal of gene therapy, the impact it has on patients, there's been large incentives to accelerate this modality. These incentives have come from drug makers, regulators, and investors. And they have led to accelerated pathways for approval, potential for expansions earlier than many other modalities, a probability of success in terms of development. The risk that one takes early versus later in development is lower, and it's increasingly dropping because of the experience we gain, and it's lower compared to other modalities. So that means faster approvals, earlier access because of these faster approvals to the need to commercialize and the preparation for commercializing. And this brings us back to OXB. CMC that needs this manufacturing piece needs an early attention. So in the next slide, 2 brief vignettes in terms of support, and then I'll round off. Here is that probability of success that I referred to. Compared to a small molecule on the right, there's these relatively modest but real increases of probability of success, particularly early in the pipeline. You can see, for example, a CAR T cell has about double the likelihood of success all the way to approval compared to a small molecule. And that we know why that is. It's because we use tweezers rather than hammers in this modality, we know the biology of disease that we target better than often with a small molecule drug, and we're learning as we're going. So in the next slides, I'll come back to some of these regulatory pieces and the importance of manufacturing, that manufacturing or that CMC piece. Regulators used to go around in meetings and make statements like 50% of a gene therapy drug is CMC. And that's not the case for a small molecule. That is the case for a gene therapy because of the complexity of the drug compounds. And when you ask sponsors, here in a survey organized by Jefferies and published last year, sponsors see as key execution risk 2 areas that directly land back into a CDMO, product quality and CDMO. These are critical paths in terms of quality and in terms of timeline for any drug development, and I can testify to that from my own experience. Now, as a last slide, in the next one, what is so important? So in the next slide, what is so important in terms of what a CDMO delivers? It's quite a lot. We're asking the CDMO to really optimize parameters in terms of speed, in terms of scale, in terms of robustness, but conversely, we also expect them to be flexible. And the only way to achieve that really is to do that with a deep level of specialization. And a party like OXB has a track record of a level of focus in the cell and gene therapy space that now goes back, I think, more than a decade plus. So with that, I'll leave you here, and in the next slide, I ask for questions that I'm happy to address in the Q&A session a little bit later. But thank you for your attention.
Thank you, Professor van den Berg, for this valuable scientific and industry context. A clear takeaway is the importance of manufacturing for the success of cell and gene therapies. Speed, scale, robustness, flexibility are key enablers for these therapies to reach the patients. And that's exactly where our next session is going to take us. I'll hand over now to our Chief Innovation Officer, Dr. Kyriakos Nitrofanos, who will explain how innovation can help us to address these challenges, how we can create competitive differentiation and enable the next wave of therapies.
Thanks, Aurélie. Thanks, Aurélie, and thank you, Luc, for that very insightful presentation. So I'm Kyriakos Nitrofanos. I'm the Chief Innovation Officer at OXB. I've been working in cell and gene therapy for over 30 years, and I manage a team working in innovation, in vector engineering, cell engineering, process development, AI, and analytics. Over the next 15 minutes, I want to explain why innovation is not a nice to have for a cell and gene therapy CDMO. It is critical for our future success that we stay at the cutting edge of this growing and exciting field, as you've just heard. So why is innovation critical for a CDMO? Well, the technology is still developing. Unlike biologics, cell and gene therapy processes have not yet been commoditized. Making vector to the scale that we need and to the purity that we need, this is still an ongoing challenge. The regulators are continuing to raise the bar in terms of the purity and quality of the vectors that are generated, and clients' needs are more sophisticated. In the past, they were expecting a CDMO to provide all the different technologies and processes, but increasingly pharma companies have got internal capabilities in this area, so they are coming to CDMOs to obtain additional capabilities that they don't have internally. And finally, it's not developments in cell and gene therapy have not finished. We hear about in vivo CAR T. We have heard from Luc on AAV targeted vectors, gene editing. All these different technologies are coming in. If we don't innovate in these areas, then we won't be able to be part of those developments. So we look to see which areas we should innovate in. We talk to clients, we talk to industry leaders, and we identified these 5 key areas. The first one is titer. So how much vector do we make in a batch, the yield? The more vector we can make, the better the cost of goods, the better the cost per dose for that particular product. The next one is speed. Clients want a batch made and released as quickly as possible, and they want that batch to be as affordable as possible. Then robustness. For every new transgene that a client will bring, can our processes generate high quality, high amounts of vector? Can our platform achieve that? And finally, quality. When we are making batches again and again, do we meet the specifications? Do we meet the titer expectations and the impurity profile to allow those batches to be released and used in the clinic without interruption? And in terms of the two vector systems that, as Luke mentioned, we are focused on Lenti and AAV. In terms of titer and speed, we are best in class. In terms of pricing, we are competitive. In terms of robustness, top tier, and in terms of the quality of the vector that we generate, again, best in class. Now, the reason we have those achievements is because of the innovation we've done over the years. These are some of the technologies we've developed over time. I'm not going to go through the whole list. Just to name a few, so Tetravector, this is our fourth generation lentiviral vector system. It has increased capacity, it gives higher titer, higher expression, and has additional safety features. For AAV, we have our Innovate system. This uses a dual plasmid production, a simplified version, to allow better ease for production, and it's married to a downstream purification process that allows us to have best-in-class AAV full to empty. We're getting 90% full from this particular technology that OXB has developed. So where do we see cell and gene therapy going in the future? Well, there are four main areas. We have AI and automation. How can we use digital tools to improve how we work and speed up our processes? Next, as we've heard from Luke, AAV targeted vectors. These vectors are targeted to specific cell types or tissues and away from organs that are sensitive, such as the liver. This opens up new avenues, new therapeutic opportunities. Next, we have lentiviral vectors used in vivo for CAR T therapy. So CAR T therapy, autologous CAR T therapy, has been very successful, but it is cumbersome and complicated. Patient's blood is taken, cells are purified, they're shipped to GMP facilities that are limited in their number. Those cells are turned into CAR Ts with viral vector and then administered into patients that have been often lymphodepleted. This means that only a subset of patients can benefit from this. If we can instead switch to an in vivo lenti, so a vector that has been engineered to only bind to T cells after administration into a patient, then it becomes an off-the-shelf product and many more patients can benefit. And finally, we have new modalities such as non-viral vectors, LNPs, that can mediate gene editing and transient expression. They can achieve things that viral vectors they can achieve therapeutic benefit that viral vectors can't. For example, repeat dosing and delivery of larger payloads. So in terms of our digital tools, at OXB we work in a number of different areas for bringing in digital tools. I'm going to describe some of these. So we have a digitized lab analysis. So moving away from manual Excel workflows to automated analysis and real-time dashboards. We're also using predicted scale-up performance. So using the data we've accumulated over many years to understand better what are the critical parameters for our processes. So can we predict when a client brings in a new gene, a new product, what kind of titer we'll achieve, and what level of impurities will be are likely to we see in that product. This allows us to condense the development time that we do in the labs, and Nick will talk more about that in a minute. So I won't go through the rest, but these different digital tools allow us to achieve higher operational efficiency. It allows us to reduce the errors, the number of deviation. Each deviation means additional work, so limiting those makes us more efficient and faster. We get a stronger understanding of processes and are able to make better, faster informed decisions. For our clients, it allows faster program delivery, lower development and manufacturing risk, greater transparency, and stronger regulatory documentation. So, as I mentioned, in vivo CAR-T is an exciting new area. From a manufacturing perspective, we see three main challenges. One is vector quantity. If more patients can be treated, we're going to need to make a lot more vector. At the moment, only about, as I said, 20% of patients are benefiting from CAR-T therapy where it's available. Next, the vector quality. For in vivo use, we want to minimize the impurity profile, so reduced host cell DNA, reduced plasmid DNA, reduced host cell proteins. One other important aspect is vector particles are made, there's a mixture of active and inactive particles. Can we maximize the number of active particles so fewer particles can be administered into a patient to elicit the benefit, the efficacy from these therapies? The fewer the particles that are administered, the less it's likely that a patient will develop antiviral immune responses, which will be a key parameter for the rollout of these technologies. And finally, vector complexity. So these vector particles will have proteins on their surface for specific targeting. They'll have proteins that allow immune modification, cloaking, and also a T-cell activation. And at OXB, the technologies that we've been developing over the last few years can address some of these needs. So we have a 400-liter large-scale perfused bioreactor. This gives us higher titer with reduced impurities. We have additives that improve the potency, make more vector particles and more active particles. And we have a lot of experience with the different envelopes that are likely to be used by clients for in vivo CAR-T. So what we've seen a surge in clinical trials in product development for in vivo cell therapies. As of March this year, there are 112 in development. 74% of these are in CAR-T. 33% are using lentiviral vectors, and 47% are using LNPs. LNPs are being used primarily for autoimmune disease, and lentiviral vectors are for cancer. Interestingly, many of these are still in the preclinical or discovery stage, 55 and 34. This means that it's going to be a few years yet before these are commercially successful. So OXB is there to help be part of that. And finally, the non-viral space. So as a company, we have some experience in this. So this is a rapidly growing modality. Clients are not viral vector or non-viral anymore. They tend to be both, and they choose CDMOs based on the ability to deliver for both of these. The clinical data that we've seen for using these modalities has been compelling. And OXB capabilities, we have experience with viral vectors, viral-like particles. Our GMP facilities, our processes, our capabilities in process development, analytical development are readily transferable to these non-viral systems. So it's an opportunity for us to enter new markets which helps protect our revenues. It de-risks cannibalization and allows us to secure new partnerships. So in summary, I hope I've shown you that innovation for a cell and gene therapy CDMO is a must, and that we've established innovation processes and governance that we're doing essentially the innovation that matters to clients. We have a culture of scientific excellence, and our scientists are very skilled in terms of the biological processes underlying vector production and purification, and are able to solve the problems that clients bring to us. We have an unrivaled track record in innovation. We've got a strong intellectual property position and deep technical expertise, and this creates a competitive moat across the company. So that nicely brings me to an end of my section, and now I'll hand over to Nick, who will tell us about how we help clients help patients.
Right. Thank you, Kiri. It's a great reminder of how important innovation is to our success and also the success of our clients. So I'm quite new to this, so for those who don't know me, I'm Nick Clarkson. I'm the head of process and analytical development at OXB. We kind of term it as development services. Now, the group consists of about 100 scientists and process engineers, very client focused. We're often the very first team that a client, once they're onboarded to OXB, they make contact with us, and we actually guide them from those very initial early stages all the way through to whatever outcome, usually commercial applications and licensing. So before I go through the presentation, because I'm new, I was going to tell you a little bit about my background. I've been at OXB for 12 years. I have worked in innovation, I've worked in analytics, and most recently I'm in process development. Sounds like a long time, but it's a great place to work, very inspirational, especially when you see the impact that you have on patient lives. Prior to that, I was working in immunology, analytics, and I spent many years in Oxford University studying immunology and T-cell biology. So it was a great grounding for the kind of work that you've been seeing today. So I was going to give you a little bit of background about process development and analytical development, where it really sits. It kind of sits between innovation and manufacturing. So it's where the commercial journey starts, really, with the client. So we take clients on board. We can do feasibility studies. We can do all the preclinical development, and this can be anywhere from construct, plasmid design, cell line development, even cell banking. And we follow the client through phase 1, phase 2, optimizing the process all the way up to phase 3 in commercial. And the important bit at phase 3 in commercial, we actually perform analytical method validation and process characterization, which is part of the process validation. And this is something that we need to do before they apply for a BLA or an MMA and get their licensing. So what really makes us unique? It's going back to this 30 years of experience and the depth of knowledge that the team have. So combination of innovation and process development, we have amazing in-house expertise in virology and molecular biology, and I think this is probably unrivaled in many of the CDMOs, and this is really born from product development, transitioning into the pure play CDMO. We've retained that knowledge so we can actually help our clients develop their vector and their product and offer advice on how to get the best results. We have a great small scale development for optimization, and this is really where we can try out our new technologies from innovation, so we can tailor it to how for a client need, so we can try different sort of enhancers or vector backbones, really just to optimize depending what their target product profile is. We have qualified scale down models. Now these are sort of smaller versions of what you'd see in the manufacturing, so we can accurately determine how it's going to perform in a manufacturing setting, but without having to do the very large scale runs, which are very costly and expensive. We leverage a lot of our data because we have a very robust and consistent platform across AAV and LV. So this is really a great benefit to our clients, and I'll explain a little bit later how this helps them out. But really key to this is the collaboration and how we work with the clients. It's central. we're almost an extension of an R&D team of our clients, and we can be very flexible and tailor our approach according to their needs. So an example of this really is, not one size fits all when it comes to process development. We have different clients. They're very diverse. Some are very early phase, small biotechs. So responding to that market need, we have a faster GMP offering. And this is a sort of a fast track route. It's leveraging a lot of our data that we know. Our platform, there's minimum development. We can go from a very small scale, amber to like 250 mils. We can go straight up to a pilot to 50 and then into GMP. We can do this in a relatively short time. And this is really leveraging our knowledge of the platform. It may not be the fully optimized for yield or titer, as we call it, but your quality is going to be good. And it's the quickest way that a small biotech will get it. And to them, quite often, time is everything. They want that first clinical batch very quickly. Kind of in the middle is like a de-risked GMP, where we'll do the small-scale studies, and we may do an STM where we can optimize some parameters. This is really trying to halfway house, getting improvements in yield before we really go into the sort of pilot and the manufacturing. And the last one is the full optimization, and this is really most popular with the well-established pharma and biotechs. We can do multiple rounds of development, so we'll do the small-scale development. We can move up into the scale-down model, the 10-liter. We take that data, and they may want to go back to the small scale and try different things. So it's kind of an iterative process. The benefit really of the full optimization for the big pharmas is they can take that data forward with them when they go to commercial, so they'll have a plan, long-term plan of how they want to get to the commercial, and they can use that data later on in their process characterization. So this is just an example of the faster GMP offering, and it really gives you a sense of how we can cut down from our standard timelines. So for the LV, a standard timeline can be 16 months. This is from a client initially making contact with us, signing up until they get their first GMP batch. Leveraging that knowledge, leveraging our data, we can really take that down to 9 months. So it is very rapid indeed, and that's releasing the batch. For AAV, it's a little bit quicker. It's quicker because the processes are slightly different between AAV and LV, but we can really get that down to sort of 7 and 9 months, which is industry leading really in that time. Some of it is skipping engineering batch, but the common theme about this is our experience coming through though. We know what are the critical parameters. We know what we can not take shortcuts, but we can accelerate and still give that same quality to the client. So going faster GMP is just one thing, but with our knowledge, we can actually deliver innovation quicker. So coming from Kiri's team, this is an example of innovation. So we work so closely together, the teams, that we know exactly what's coming out of the innovation department into the process development. Now, this is an example of a change that we did to our downstream processing. So you probably all appreciate that yield is everything. And historically in LV, it was quite low. We had a new downstream processing. It's an ion exchange resin. And this increased our yield about threefold. So you're getting 3 times as much product at the end of your processing. Now, it was in innovation, and we were challenged by a client. Can we adopt this technology very quickly? They had a very important program. So we took it from innovation through process development into GMP in 6 months. This was an amazing feat for us. The actual results, these are real results. It was a threefold increase. There was improved quality of the vector. And what it really meant for OXB is that we gained the trust of that client, and we gained their business for a key client going forward. How do we do it so quickly, though? This really comes back to the innovation and how we employ that in our development and in our manufacturing. So we use statistical approaches to determine how inputs affect our process. Now, this is what we call design of experiments. It's kind of a very basic AI for people that don't know. AI really is just very advanced statistics, but the design of experiments, it uses statistics to accurately predict what's going to affect our process. So what this means, we can study multiple parameters or inputs, we call it, at once. So doing 1 or 2 is quite time consuming in a lab. We can do 10, we can do 100, and that's by utilizing the automation that Kiri was talking about earlier. So we can do multiple inputs simultaneously and really drill down and get a better understanding of how this technology is going to improve us. What it means for the clients, fewer experiments. We can do things quicker. We can leverage certain software and programs to sort of better predict what's going to happen. You get faster onboarding and progression to process characterization, faster to GMP. But particularly important really is the robustness of these automated processes. When you're doing these kind of development works, if you're not robust, the development kind of means nothing, and we're very good and we're very robust at doing that. And what it means really for OXB is we can service more clients, and it really reinforces us in our market leading position going forward. So we talked about faster GMP and we talked about innovation. Another really important thing for us is our de-risking that path to commercial. And I think this is one of our real strengths. Our in-depth knowledge and our experience really shows through when we're working with our clients. What I mean by commercialization is taking the product from the sort of development phase and validating that process so that they can go for a biologics license application or a medicines marketing authorization. We do this very well. We have the previous slide you saw, we've done about 10 of these process characterizations, which I think is probably more than any other CDMO in the market for viral vectors. We have standardized templates. We have the platform knowledge that we can leverage. We have the qualified scale down models, and we have that extensive process characterization experience. What this means is that we can really accelerate this process and the clients have confidence in us. Faster development times, lower development costs, and really what it's setting OXB as is we're kind of the go-to CDMO now for these late stage commercial programs, like the go-to CDMO, because a lot of the other CDMOs do not have this experience, so it's kind of a sure bet if you go with OXB. And this is just an example of what we can do. So when you're going through a commercialization process or process validation, it's very time consuming. There's very long timelines. There's a very high experimental burden. It's very costly to the clients. So what we do at OXB, we take this leveraging historical process data where we can approach. And this is always in conjunction with the client. So we can focus on the parameters that we know to be genuine and really will impact our process. And we will exclude some of the well characterized or well understood processes. So this means we focused all of our energy on areas where the regulators are going to actually put more scrutiny. The impacts, we get the accelerated commercialization timelines. Here we've taken 6 months off of a program. That's half a year off of a commercial program, which is a big deal for a company trying to take their drug to market. It's reduced resource and reduced development costs. And it's just a more efficient way of working. But what we don't compromise on is quality. And I think this is, again, what's really setting us apart. We have that trust of the clients. They see our track record going forward, and they know we can deliver when we say we'll deliver. So just summing up, again, it's going back to this sort of client-centric model. It all comes from our track record. This 30 years of experience is really incredible. There's the unrivaled technical excellence, and deep knowledge of the vector platforms. It means we can do things that others can't in the field. We have that flexibility that you've seen. We can tailor our approach to the client. We can accelerate, or we can go more in depth for the client. And that extensive commercialization experience really is key, I think, going into these later stages. As more of the drugs are actually entering those phases, we're in a prime position to really capitalize on those. And it's shown through our repeat business. So client satisfaction is very high. So where we have clients with multiple assets in their portfolio, we tend to get those as well. So it's that repeat business, particularly going into the late phases. So I hope that gave you a little more insight into our process development and excellence, client centricity. And I'll hand back to Aurélie.
Thank you, Nick, for this clear and practical overview of process development. The message is clear. Process development is not just a step toward manufacturing. It's a critical lever for speed as well as cost and risk reduction. And it can become a competitive advantage when a platform approach like OXP's can cut time to GMP by 6 months. We'll move on now to our Q&A session, slightly shorter than initially planned, but don't worry, there will be another session. We'll start by questions from the room. If you have a question, please raise your hand, wait for the microphone, state your name and organization, and also a speaker if you have a preference. I'll invite to the seats Nick, Kiri, and Frank. And we also have Luc, who's also available for questions. Please go ahead.
Hi, this is Charles Weston from RBC. Thanks very much for taking the questions. 2, please. First of all, for Nick, you mentioned the process development timelines and the process characterization timelines, both you described as best in class. Are those, I guess, how confident are you that they are best in class? Are they key characteristics that win you business? Perhaps also to Sebastian, in terms of, how commercial that offer is. And the second question, Kiri, you mentioned LNPs quite a bit as another modality alongside viral vectors. Again, I'm not sure who the right person to answer this is, but would it be an important development for OXB to be able to offer those sorts of technologies alongside viral vectors? Thank you.
Right. Well, I'll start. We are best in class when it comes to the commercialization. We've done 10 of these process characterizations. Not all of them have gone to commercial yet. Some are in progress. Some are, other things happened. We've never had negative feedback from a regulator or a client on our approach to these process characterization and that whole approach. So I really do think we are best in class, and we do get the repeat business from clients that have gone through the process with us, and they're putting more assets our way. I don't know if Sebastian wants to add.
Yeah, so that on? Works. Yeah, thank you. So talking about the timing, how critical is that to win business was your question. The experience in process characterization tells you what you must do and what you can skip because we have the data coming to the platform. You know that because you've done it not just once, but 3, 4 times. You've discussed with EMA, MHR, FDA, and they tell you that's fine. That can come from the platform. You can skip that step, and this set of data you generate. For the reason that Nick just mentioned, we've done that 10 times. One of the phase 3 programs on which we were approached 2 years ago, the question was, how fast can you do that? And the standard timeline is usually between 18 and 24 months. And they said, if you can do it in 12 months, we work with you. We don't even question other CMOs. We go with you directly. We started the onboarding 12 months after we had delivered the GMP batch. We had used the platform data for the discussions with FDA. Went very well. It's part of the 10 now. Soon it's going to be history and waiting for the final approval. And we'll tick the box of another commercial product with OXB very soon. So it's critical to the point where people can come to you and say, you've done it before. Can you even be faster? The answer is yes. Because of automation, because of new tools coming from innovation, and again, because we've done it before on time in full. So it's indeed critical to the business.
And perhaps let me add on this question about best in class. I like your answer, but that's the answer which we get also from our clients. So it's not coming out of the blue. It's really what clients are telling us. And this is coming back to the 30 years of experience that we bring on the table. Yeah. And because we just focus on viral vectors, we just do that. Yeah. We are probably able to deliver the best service possible.
With regards to does that answer your first question?
Yes.
With regard to your LNP question, so we are now getting clients who have both modalities, and they're asking us, you've done a great job on the viral vectors. Can you help us with our LNP need? And we're seeing in terms of the new products coming through a mixture. So sometimes you're seeing a combination of LNPs and lenti, AAV and lenti, viral-like particles. So being able to be in both viral and non-viral should allow us to help these products that are a combination as well as viral vector pure or LNP.
So he's our Chief of Innovation, so he's ahead of us, yeah? But I want to mention, for now, we do viral vectors. It's fair to say, Kiri, that we look at non-viral, but probably not in the next 6 to 12 months for sure. But perhaps, Sebastian, you can address some point in your presentation later on where we are with non-viral.
Thank you.
Hey, good afternoon. It's Miles Dixon from Peel Hunt. If I can just return to Kiri, it was the grid that you presented on the metrics, the quality, the yield, the pricing. On pricing, you said competitive. Is that competitive adjusted for the quality and the yield and the titer, or is it just competitive on an absolute basis?
I'll refer that to Sebastian.
Thank you, Kiri. So when we're talking about competitive, Kiri's example was about what we would consider the final product. In the end, the cost per dose, which is the cost per patient, must be competitive. I'm not just in charge of the business activities. I'm a scientist myself. So comparing data to data, I would say if we look at the cost per dose strictly, we're very competitive. If I look at the prices of the services we offer, we are indeed competitive. But the prices of the services we offer include the process development activities, the manufacturing at clinical scale, the manufacturing at commercial scale. The cost per dose is only commercial manufacturing, where we enjoy some benefits of the development through the robustness of the process. Process development we have a lot of uncertainties that we face. I mean, sometimes you have to change. So you understand what a budget should look like, but you will see variations. On commercial manufacturing, you don't see variation. You run a batch and then another batch and a third batch and a fourth and so on. So we know it's robust and we know that we can deliver cost per dose that some of our clients in the recent years, I'm going to talk about the past 2 years, have considered aggressive. And we've reached the target that they fixed. And that's why I can say very competitive if we look at the cost per dose. Cost. Price is a different story.
Thank you.
Hi there, it's David at Berenberg. I just wanted to do sort of broad brush comments about whether you've seen any changes to your competitive position recently. So have there key qualities that clients have valued changed over the last several years? So whether we be talking speed versus process development versus regulatory assistance versus house QC versus geography. I'm just wondering how you've seen that evolve, please.
Yeah.
You want to?
I can take it, yeah.
Yeah, sure.
Yeah. So we've seen an improvement in all three. So one of the biggest things clients used to ask was yield several years ago. We're very competitive on the yield, but you saw from the slide, we've improved it over threefold now with our new process. And the other point was speed. Now we've cracked the yield, the speed is really becoming critical. And I think every program that who wants to take a long time to get their drug to market, so the pressures are there really for speed going forward, and that's why we're really leveraging the data and constantly improving what we can do going forward. So I think that's how we're staying ahead of the curve really against our competitors, and I think we hope to remain there going forward.
We'll take one more question. Don't worry, there will be time for a second Q&A.
Good afternoon, Kane Slutzkin, Deutsche. With therapies potentially moving sort of into earlier lines of treatment and patient populations expanding, how should we sort of think about sort of vector demand growth? Is it sort of broadly linear to patient growth, or how should we think about that? Thanks.
Very good question, Sebastian. You are the nearest to the client, but it's an interesting question.
It's a difficult question, actually. Would love to have Aaron's point of view on how the dynamic is evolving. But seen from my window, it's not linear at all. Because I would say first, we are not working in a global environment from a regulatory standpoint. If you file with FDA, you file for USA. If you file with EMA, you file for Europe, excluding UK now. So you have to file with MHRA to be in UK. And we're working on drugs, commercial, approved in US, where our clients are looking at some countries in Middle East, some countries in Asia, some countries in Europe, not even all of Europe, which means that you're going to see the progression by step. It's approved in one country. And let's say that because it's one of these countries where at the very same time they grow their biologics and cell and gene therapy markets, it's going to be adopted very quickly as a second line of treatment, for example, when cell and gene therapy is not, as a standard, a second line of treatment in the rest of the world. Most of the time, it's a third or fourth line of treatment. So for me, it's not linear. It's really a stepwise approach. If it was truly global, if we had alignment between the countries and the regulatory bodies, that could be, but we're not there yet. Well, we'll discuss that question later, Howard.
So we're taking one question from the room. So how do you balance the inherent bespoke nature of each client program you mentioned to maximize safety and other metrics with the needs for a more commoditized offering that drives the lower cost of goods and hence higher margins?
I'm sorry, Sebastian, but this one is for you.
There's a second part. You want it now? If process intensification sustainably improves yields, how much of the economic benefit is retained by OXB versus passed on to the client to improve that competitiveness you have discussed?
Gosh, where shall I start? I'm actually going to take it as one question. There are solutions today to go extremely fast to a first batch. Does it mean that all our clients are going to accept that solution? The answer is no. So when we talk about the cost of developing and the cost of manufacturing, you need to look at these costs through the lens of each client objective. Some clients from the very beginning say, I want a cost per dose that is the lowest possible. And Nick will tell you that and Thierry actually will tell you, if you want to go to the lowest, you need to develop a stable cell line, which is part of the efficiencies that we discuss in the second part of the question. It's going to take more time, though. Is it going to take more budget? If you take the overall process development budget, not really. But it's going to take another 6 months to 1 year, depending on the difficulty of creating the cell line. But the cost per dose in the end is probably going to be divided by 3 to even 10 if you decide finally process intensification to run not at 200 liter, which is more or less the standard in gene therapy today, but you decide to run at 1000 liter, which we can do as well. So if you combine from the very beginning in your strategy, the cell line development and the final scale up, we will pass all these benefits to the client. And I will come back on the why in a minute. We intensify the process. We run at a larger scale. It's not increasing significantly the cost of process development and early stage manufacturing, and the benefits are huge in the end. Why do we want to pass these benefits to the client? Because, and Nick said it in one of his presentations, the client was very happy with what we delivered. Now we have recurrent business with this client, exactly for this reason. If the client is happy with us, we'll get program number 2, number 3, number 4. And when the fourth line of treatment will become third line and then second line, all that volume will be with us. Which means that if passing the efficiencies to the client is seen by many analysts as we're losing value for OXB, no. Midterm, there's a huge value for OXB, and we want to capitalize on that value.
Thank you, Sebastian. Thank you to everyone who asked a question, and thank you to our speakers. We'll now take a 10 minutes break. See you back there at 3:30 sharp, so we are not late. Thank you.
Welcome back. My name is Sébastien Ribaud. I joined the company 3.5 years ago as Chief Business Officer. spent pretty much 15% of my career on the technical side, starting as a gene therapy scientist. Before what Luc presented, first casualty in gene therapy in 1999, that pretty much killed the efforts in gene therapy during 10 years. Good to see that the field is back. And the last 15 years of my career on the business side, including activities with a fairly large services business where I was heading a business unit in charge of cell and gene therapy and biologics. I'm going to drive you today through our pipeline, how we grew our client base, and tell you a bit about how we convert our proposals into a real contract and how that is translated into the day-to-day activity at OXB. It's going to be the first part of my section, and then I will tell you not only why the clients, how the clients choose a CDMO, why they choose OXB, why they stay with OXB. But my opinion may be interesting, but it will be much more relevant to have Arun's opinion through the fireside chat that will follow immediately my presentation. Frank presented the space in which we play, viral vectors. And we often associate viral vectors and cell and gene therapy, which is wrong. Cell therapy is one area where sometimes you're not using vectors, which actually you will see here on that slide as cell therapy. We use a vector when we modify the cells. You can also use non-viral vectors to modify the cells. They are flagged here as only one segment, the RNAi gene therapy. So we play in a field that is relatively complex when we say CGT. We actually play in gene therapy, including the gene-modified cell therapy. And if you look at that field, I often read, including actually this morning, comments saying the field is depressed, the field is growing, but it's much slower than what it was in the past. Well, I would actually disagree. We still see the field growing 35% year on year, as you see it there. And we play in three segments: the gene-modified cell therapy, the gene therapy, and the oncolytic virus space where we play. As you see, that's the majority of this business. And here, it's not just the viral vector numbers that we're looking at, just to be clear. It's the entire field of cell and gene therapy, but fast growing market very, very clearly. Now, if you look at the viral vector space only, there's often a question, how much are you afraid of companies investing in their own capacity for manufacturing? We are not. And you see here the trend. 2025, we estimate, well, the market studies estimate that 75% of the activities are outsourced. If we go to 5 years from now, we estimate that up to 80% of the activities will be fully outsourced. Why? We talked about it just before, cost of goods. If you want to make sure that you have a cost per dose and cost of goods that are low, you need to use the facility at their max capacity. If you run one program only and you make 10 batches per year, there is no way you use a facility at its capacity. You need to be manufacturing hundreds of batches per year, and even regulatory agencies like FDA have recommended that the biotech and pharma of this world work with CDMOs to make sure that the capacity is used and that the costs are going down. So it's a very complex field, as we know. Reason why my colleagues emphasize the 30 years of experience of the company. You are successful in manufacturing only if you've developed the right process. It takes a few years to have this experience. If we go now into the field in a bit more details, similar to the comment on the field is depressed, I hear many times, what about funding? How's the funding situation? Are you afraid that these programs won't progress? Well, factually speaking, comparison between Q1 25 and Q1 26, overall the number of programs in gene therapy here is stable, 2,100 programs. What has changed, though, is that the preclinical stage, where you run feasibility studies. You check if your gene of interest combined to the vector has the effect that you expect. This segment is indeed going down from 1,400 to, let's say, 1,300 here. But the other segments, phase 1, phase 2, phase 3, and pre-registrations are the ones that are going, which means that the field is gaining in maturity. And that's the reason why we've seen in the first years of gene therapy very few programs being adopted, and now we see 5 to 10 programs being adopted every year, which has a big impact for CDMOs. It means that the routine manufacturing is needed. It means that the process characterization capabilities and capacity that Nick mentioned earlier, where we make sure that the program is robust, that is needed. Which means that we're running more phase 1, more phase 2, and more phase 3 batches. And for a CDMO, that's exactly where our business should be. We should be developing programs where the feasibility has shown that we had valid candidates. We should be manufacturing the batches and making sure that these products become commercial and are going to be accessible to patients. Gene therapy is about 50% of the entire field, and if I look at the past 3 years, it's been pretty stable. There's often that question, are you afraid of a complete replacement of the viral space by the non-viral space? Well, actually, RNA is here, and it's been pretty stable as well. So they coexist, but we don't see one replacing the other. That's why we believe that viral vectors have many years in front of them, although, as Kerry mentioned earlier, we're looking into the non-viral space to be ready to invest when the time will come. Moving from the market to the Oxford Biomedica pipeline now. 3 years ago, we were a lentivirus vector company, and the pipeline was 90% lenti. We had a few AAV programs, a couple of adenovirus opportunities as well. If you look at the pipeline as it is now on the left side here, you see that for the very first time in 2026, we have even more AAV opportunities than lenti. Result of the one OXB strategy where we acquired experience, expertise, and a platform in the AAV space when the company acquired the site we have in Bedford, Massachusetts. We've developed our customer base in AAV, and that's why today we see on par AAV and lenti. We've also made sure that we were diversifying the pipeline in terms of clinical phase or commercial phase where we work. And you see that we have almost 20% of our activities in terms of distribution of opportunities in the commercial space and 11% in phase 3, meaning that about 1/3 of the opportunities on which we work today are revenues with recurrent manufacturing, phase 3 preparation of the commercial introduction and commercial manufacturing as well. We still keep about 1/3 of the activities in the preclinical space because we want to make sure that we support our existing clients who have new programs that they want to develop, and also emerging biotechs or small biotechs who come to us saying, we have a candidate, we would like to run the feasibility with you, because we know that when they run the feasibility with us, they test our platform, they see the benefits in timing, in automation, and so on, and they stay with us for the other phases on which I won't elaborate here, phase 1 and phase 2. The biggest change we've seen in the past year is the distribution between the geographies. Back to 2022, we had 90+% of the activities in Oxford, UK. The rest was in Bedford, Massachusetts. With the acquisition of Lyon and Strasbourg, you see that today 20% of our opportunities are in France, between the two sites here or directly on our site in Lyon there. And you see that the US also have a very significant presence now. I remember the time when we were discussing 90% of opportunities in Oxford, 10 outside of Oxford. Today is a very different picture, 70% outside of Oxford, 30% in Oxford. One of my colleagues recently asked a good question, which was, does it mean as we have less opportunities for Oxford that the absolute value of the pipeline for Oxford went down? Absolutely not. because at the same time, we were growing the pipeline value, and the pipeline value has more than doubled in the past 3 years. So in absolute value, we still handle the same volume of opportunities for UK, and we have filled the other sites with new opportunities. If I look at the distribution over just 1 year of the pipeline dynamic, it's very interesting to see that in Q2 2025, we had 13% of the opportunities for the French sites. We still had 67% for UK, and the rest was US, Bedford exclusively at that time. If I move now 12 months later, Q2 2026, 19% in France, so obviously increased versus about a year ago, 46% in value here, not number of opportunities that we had on the previous slide. 46% of the value of the pipeline is in Oxford, and the rest, very significant number, about 30%, is for the US site. Why such a dynamic? For one reason, the acquisition of a commercial-ready FDA-approved site in the US, which was needed, which was requested by our clients, and which had an immediate impact. We have Thursday this week, in 2 days, a kickoff meeting of a phase 3 project in Durham, North Carolina. The process characterization will be done in Bedford, Massachusetts. The phase 3 batches, the process qualification of the manufacturing process will be done in Durham. The same clients, without the acquisition of Durham, we could not have executed in the US. So the acquisition of the US site completely changed the pipeline dynamic by site. And if we look only at the first months of the year here, we built that site about 3 weeks ago. The number has changed now. But we had signed at that time 10 new clients, 4 coming from US, 5 from Europe, and 1 from Asia. Actually, we have 1 team in Asia today working with the client on the initiation of the program. Let's go back 3 years ago. I would have said 7 to 8 new clients in the US, 2 to 3 in Europe. That was the situation. With the One OXB strategy, we're really recognized as a global organization today, attracting some clients in Asia who did not even know that OXB was a CDMO at that time, attracting clients who understand that we've run these 10 process characterization and validation, and that we can do the same for them. And that's why we acquire clients at phase 3 now. That has been possible only because of the expansion of the organization, including the new vectors, including the new sites, and including some recurrent revenue that you see here in existing business. When one of our existing clients tells us, "I would like a proposal for a potential new project," 84% of these proposals give a contract. That explains why we're very comfortable with our customer base, because usually when they tell us, "We have a new program, what do you think?" in more than 8 cases out of 10, they sign with us. If we're at the negotiation stage, meaning it's not just a proposal on 1 page, that's the timeline, that's the budget, it's a real negotiation, we sign 97% of these contracts with existing clients. Moving to people who have not worked with us before. If we issue a proposal, again, 2 slides, 1 timeline, 1 budget, we sign 1 program out of 3 at proposal stage. If we've drafted the contract, and we're not negotiating the terms yet, if we've drafted the contract, meaning we have a scope of work, we're signing 2 out of 3, 72% exactly, but we sign 87% when we're at the negotiation stage. I've worked for 3 different services business. That's the first time I see conversion rate that high. That goes with the customer satisfaction that we enjoy, which on a scale between 1 and 10 is systematically above 8. If I take the average of the responses we have from our clients, to whom we ask twice a year to fill a questionnaire. How did that change the situation at OXB? You see it here. The number of preclinical development and early stage, meaning phase 1, phase 2 activities, has been pretty stable between April 24 and May 26. And I'm going to focus actually on the difference between March and May 26 after. But number of early stage program pretty stable, between 40 and 45 roughly. The biggest change that we've seen in the last 2 years is the increase in late-stage program. We doubled the number of late-stage programs from 3 to 6. We added 1 more commercial programs. And I'm looking just at the dynamic in the last year from 4 to 6, from 2 to 3. And in the last 3 months, from 5 to 6 on late stage. People come to us because they understand we're a CDMO who can manufacture in 2 different geographies commercial products. So we can ensure dual sourcing. Do we face tariffs? No, because we can manufacture in the US. Can we give you access to tax incentive in Europe? Yes, because we can make the manufacturing in Europe. And so far, the Asian customers have been pretty happy with us making the manufacturing either in US and Europe. I'm going to finish very quickly on that one. The pipeline is also divided between the different client segments where you see emerging biotech, established biotech. We would consider Cabaletta Bio as being an established biotech. And finally, you have the big pharmas here. It is as it should be. We want to support early stage activity. We are not a CMO or a CDMO. And we want to continue innovating and developing for people who need support at early stage. That's why the emerging biotech segment will stay high. I'm going to move now to the second section, on which I will be quick because I believe that the discussion with Arun will be better than just going through slides. But why do we see clients selecting OXB? And first, how do people select a CDMO? The selection criteria are more or less always the same, and I've personally not seen any variations in the past 5 years. Track record is number 1. How many times did you do it on time, in full? How many times did you respond to the regulatory agencies successfully? Could you take a program at early stage and push it to late stage? Do you have the technical expertise and experience, which is slightly different from track record? Do you have the slots? Can you make it now? Can you start? Can you be fast enough? Can you manage complex programs? It is very complex. You need to design a vector. We're not working with just one vector, but multiples, lenti, AAV, adeno, MVA, you name it. The list is quite long. We have this vector know-how. We have the manufacturing capability and capacity. We have the experience to support end-to-end. We can work on hybrid capsids where you need to create a new capsid. We can work on the stable cell lines, as we discussed earlier. So that's very, very early in the development process, and we can bring these programs to the commercial stage. Last but not least, and for me, that's one of the most important points here, we don't want to be seen as an organization that is only dealing with clients' transactional relationship is the way I would call it. We're really the development and the manufacturing arm of the companies who come and work with us. If you speak with our people running a project, they don't talk about the client project, they talk about their project. And we're also very impacted when we see that clinical data are not that good because many of our people, if not the vast majority, are motivated by giving access to new treatments to patients. So that's why we believe that each relationship can be a strategic partnership. And I like the question, aren't you afraid to be exposed to emerging biotech funding issues and so on? But some of these emerging biotechs are called today Gilead, Sanofi, Novartis, BMS, and so on. But they started as a very small company as well. So we are working today with the future BMS, Novartis, and so on. Why do they come to us? They come to us to de-risk the path to approval. And I won't go through all the points because these points have already been detailed by Nicky in his presentation. We develop for GMP excellence in the future. We want to make sure that the process are robust, they are validated, and we're going to deliver a consistent result. Then comes the quality and regulatory part. The documentation is ready. We can manufacture according to ICH, to the international quality standards. We're ready for any regulatory inspection, which means that we provide regulatory support to the client for the filing, and we're ready to be inspected by the agencies before the introduction of the program on the market. not only we want to attract clients, but we want to retain them. And I must say that since I joined the company, we've not lost one client who said, I'm leaving and going to another CDMO. Why are the clients staying with us? We try to extract from different customer survey the main arguments that we heard coming from our clients. The first one, successful tech transfer. We made a feasibility somewhere else. Can you take that in your lab and make sure that you optimize so that we have a better process in the future for early stage activity? Same comment for people coming to us saying, we were working with a CDMO that's made a phase 1. They don't have the experience for phase 3 and commercial. Can you do it? We did it successfully. I talked about the 12 months process characterization earlier. I'm not going to come back on that. But we have a very structured transfer support. If we ask people, what do you like most with our scientists? Responsiveness. So always number one, you guys propose options. You provide solution. We come with a problem. You're extremely responsive, and you try to find a way to unblock the situation. Once we've defined the process, we deliver in a reliable way. It's consistent. It's on-time execution. OTIF, as we call it, on time, in full. And last point, which gives me the opportunity to ask Arun to join me on stage, long-term trust. We want to be seen as partners. It goes beyond just delivering the development or just delivering one batch. We want to be seen as the commercial partner for the future of the program and of the treatments we're going to put on the market. Arun, if you don't mind joining me.
Thank you, Sebastian.
Since it was a long monologue, I'm going to pause for a minute and ask you if you don't mind giving a few words on yourself.
Yeah, absolutely. Thank you, first of all, to you for the opportunity to have this chat, to the Oxford team for inviting me here. It's a pleasure to be here. My name's Arun. I am Chief Business Officer at Capilleta Bio. Been with the company since its founding in 2019. My background started off as an investment banker. Went to medical school, was a pediatrician at the Children's Hospital of Philadelphia, and then came to Capilleta in 2019. So I've been there for 7 years now and oversee our business development, enterprise strategy, and new product planning efforts. Capilleta as a company, just a brief bit about Capilleta, was spun out of the University of Pennsylvania and formed for the sole purpose of developing and launching the first curative cell therapy for patients with autoimmune disease. We started off with a legacy platform and evolved into CD19 CAR T therapy about 4 or 5 years ago with the onset of the initial academic data, and now have a program in registrational studies and several other clinical trials in development evaluating our product candidate called Rese-cel, both with and without preconditioning. And so that's us as a company.
Thank you, Arun.
Yeah, of course.
I was actually not with OXB when you selected OXB as a CDMO, and I joined at a time where the project was still considered as an early phase project. Mm-hmm. And I said why many of our clients choose us. I mean, but I would be interested to know what are the important criteria that you list when you want to select, generally speaking, a service provider and more specifically a CDMO.
Yeah, absolutely. So one of the things that I learned very early on at Capilleta was the process is the product. And that line stuck with me in a way that few other things have in terms of being true and seeing it illustrated over 7 years of being at the company. And so by that, I mean, when you have every patient product needing to be manufactured in and of itself, you realize how important manufacturing is to the overall patient experience that's being delivered. And so for us, when it comes to selecting strategic manufacturing partners, it really is, to your slide, not about a transaction. It's not about individual features. It's about the holistic opportunity to work longitudinally with that partner over time. And so I tend to break that up into kind of 3 things. One is technical expertise, which focuses on reliability, quality systems, on-time delivery, a lot of the things that Kiri and Nick mentioned in their presentations. There's alignment on the strategic drivers of the field, what's going to move the field forward. There, I think about scalability, and innovation, because what the business is today or what the business was a year ago is probably not what it's going to be like next year, 3 years from now. And so we have to have a shared commitment to always look for how to optimize, always look for how to do better for patients, to do better from a cost of goods perspective, a yield perspective. And that's something that if those two parties don't share a view on, it can be hard to operate. So that's certainly important. And then finally, I would say cultural fit, which is you can spend weeks or months, writing out a contract, negotiating it. And another thing that I've learned since I've been here is if you have to go back to that contract after you've signed it, it's probably not a good thing. And so I can say the teams, both at the leadership levels, the operating levels, every kind of function that I can think of has the ability to reach out to their colleague on the other end with a call, with an email, and work collaboratively to help solve whatever the question or issue may be. And I can also reflect a lot of the things that you summarized in the survey, which is the responsiveness of the team and the commitment to wanting to create a solution that maybe we hadn't even thought of to begin with, is something that over the 7 years that we've been working together has really been noticed by the team.
Do you remember what was the most differentiating factor between OXB and the other CDMOs at that time when you made the selection?
Yes, so we've actually been working together. We're one of those repeat clients that you talked about because we've been working together since our legacy platform back in 2019, where we initiated some pilot work with the company. We worked on that together for 2 or 3 years. And then when we pivoted to Rese-cel, our CD19 program, continued working with Oxford. At the original 2019-2020 decision, the primary driver was reliability, ability to technically execute, and the fact that you all were really the only ones that had done it before. That was, I would say, sort of the initial framework, in addition to the same strategic alignment on views and the cultural fit that we had noticed. The years that we spent working together just reinforced those principles. And so by the time that we had Rese-cel and we had a decision internally to make around who do we want to work with, at that point, it really wasn't a question. It was, we've had such a positive experience working with the team over time. And one other thing I'd say is that the longevity of the folks that are at the company and the low turnover rate is a really important feature of working with folks because it reflects what happens at Capilleta too. There are folks at the company, and the majority of folks that are the only people who have ever held their position at the company. At Oxford, we see a similar feature of low turnover, consistency, and interaction. When you know the people that you're working with on the other side, it really becomes an easy decision.
Oh, yeah. So much easier.
Yeah.
And actually, I must say you're absolutely right. Since I joined the company, I've worked at Capilleta with the same team.
Yes.
Including
Yes, that's exactly right. We like to say, there's a picture on our website where I think 8 of the 10 people or 8 of the 11 people, however many people are up there, are the first people to ever hold the role at the company and the only people to ever hold the role at the company in the history of the company. And so I think that is unique in biotech and something here in this case that I think is quite unique to Oxford as well, which is the continuity and longevity of the team that's there.
The world has changed between 2020 and today. Have your selection criteria list changed as well, or would you make the same selection with the same criteria?
I think the question on selection, yes, we would make I think the outcome wouldn't have changed. I do think there's been some evolution in the criteria, not in terms of adding and deleting, but more in modulating the emphasis on some of the criteria. So for us, as we've transitioned from, as you described, early clinical stage to late clinical stage, and as we've seen Rese-cel grow and demonstrate clinical data, the opportunity for patients to really benefit in a significant way, what we've realized is that the importance of scalability, industrialization, I really like Luke's word and sort of the phrase he used, industrialization of the manufacturing process is increasing in importance, because what we realize is if we truly are able to execute on innovation, we can open up the opportunity for maybe an order of magnitude more patients to be able to receive CAR T therapy in our case. Well, then we're going to have to be able to make enough product for those patients to be able to access it. And in order to do that, we need partners who can reliably deliver at scale, and reliably deliver at scale at a cost that is provides the opportunity to be able to give the drug to patients at that scale. And so I would say there's more been an emphasis in those criteria, not really a change in what the outcome would be, but in how we think about really optimizing the opportunity for folks.
Understood. I had on one of my slides the outsourcing rate, which is increasing. You made the decision at Cabaletta to outsource some of the activities that OXB is running. When exactly did you make that decision in the development process, and why did you decide to go outside instead of building the capabilities internally?
Yeah, it is a very, very insightful question, because I would say in the early years of the company, so for the first 2 or 3 years that we were in existence, this was a never-ending discussion. And by that I mean at the time, so this is now 6, 7 years ago, there had not been a cell therapy company that had been able to achieve success without having its own manufacturing facilities, both vector and cell processing. We had many, many discussions and opportunities around, should we invest in our own bricks and mortar? Should we continue working with CDMOs? Again, at the time, the ability for and the commercial experience of CDMOs, including Oxford, was much less than what it is today. So this was a discussion that, again, was really ongoing all the time. What we eventually made the decision of, realized, and are now, I think, grateful that we did, was that we as a biotech company have limited resources, limited people, and a need to allocate capital and people time to the activities that we do best. And the things that we believe we do best are clinical development, commercialization of therapies, discovery of new therapies. And if we can find trusted partners that are able to take on and partner on certain core attributes, core capabilities that we need for the product, that's a good investment of our time and capital. And the other thing I will say is that, just purely from a capital perspective, when you put in bricks and mortar into the ground, the amount of money that takes for an emerging biotech that's in development stage can often be prohibitive. And then you also get to the question of, and I think you had this in your slides as well, capacity, which is you're building a facility that you hope at peak capacity may be able to support thousands of patients a year. But when you start off as a company with a product, you're unlikely to be at whatever that peak capacity is. So for the years that you're building up to that capacity, that excess capacity is just coming down into your bottom line. It becomes hard to operate. For us, making the decision to partner with CDMOs from the get-go has eliminated a lot of those fixed costs, eliminated a lot of those carrying costs. And we're grateful not only for the benefits from a financial perspective, but because it really allows us to focus our energy on the places where we can deliver the most benefit overall.
There was a very interesting question earlier about evolution of the patient population.
Yes.
How's the dynamic and so on. And for me, there's a link to how you built and how you work on making sure you have the right commercialization strategy. Without entering into confidential information, can you tell us how you're building the commercialization strategy for what will be soon your first product?
Yeah. Here, I think there is a foundational assumption, because CAR-T has been a commercial entity now for nearly a decade. And I think we have made real efforts to try to delineate between CAR-T commercialization in oncology versus CAR-T commercialization in autoimmunity. And for us as an autoimmune company, CAR-T commercialization in autoimmunity is going to look very different. The biggest reason why, in our view, is simply the number of patients that can be served with the therapy at hand, and the benefits that the therapy is delivering, both from a safety and clinical activity perspective, are really promising and sort of point towards a scale of patients that can be provided this therapy in a way that's just fundamentally different from what we see in oncology. So I say all of that because it absolutely has affected how we think about commercialization and what we emphasize. So the things that we are emphasizing are being able to, and I know these words keep coming up, but being able to make enough products for patients, so scalability, being able to do so in a way that makes the therapy not cost prohibitive. Because if you're treating tens of thousands of patients a year, not hundreds or a few thousands of patients a year, you get a very different sort of, economic return, economic look, and you need a different profile in terms of what the product is that you're making. And so it's about innovation from being able to deliver a drug that is suitable for enough patients, and then being able to make enough of that drug to be able to give it to patients if and when they need it. So that's really been at the forefront of our commercialization strategy. And again, being able to do that with folks who understand it, who share the view of the importance of what that can mean, and then who work with us to help accomplish it, are how we are successful.
I'm going to look at Aurélie to check if we have time for one very last question.
One more.
Thank you. How important was it back in 2020, and how important would it be today to select a player that is by default a global player?
Yeah, understood. I think it would be naive to think anyone in this space can be successful by focusing solely on one country, and that how each individual company's commercialization plays out on a global scale to be determined. But I think everyone believes it is to your benefit to have a presence in multiple geographies, to advance regulatory clinical efforts in multiple geographies, and then to be able to have the opportunity to commercialize globally. And because of that, basically manufacturing capabilities to enable that is almost like the ticket to play. It's table stakes. And so we require any CDMO to be able to manufacture across geographies, because if we're restricted in one area, restricted to one area, then you essentially have to double the work that you're doing simply to expand a geography. And that's really, again, not an efficient use of capital, not an efficient use of time, and forces us to take time away from, again, the things that we feel like we do best. And so when we can work with partners who have that capability already checked off, who have already gone through the process with the EMA, gone through the process with the MHRA, gone through the process with the FDA, then those are things, it's not that we don't have to worry about it, because we absolutely would worry about it, but we know we're working with someone who's been through it before and can give us experience, guidance, and advice along the way while we're working together to advance it in whatever geography we're looking at.
Very clear. Yeah. On time.
Oh, wonderful.
Thanks very much for coming. I know that Philadelphia is not really next door, so thanks very much for making the trip and coming to us.
It was a pleasure.
Thank you,
Sebastian.
Thank you, Arun, for your perspective, and thank you, Sebastian. I hope you found the discussion insightful. What clearly emerges is that track record and experience are critical when selecting a CDMO, as well as global scalable manufacturing, quality, commitment to innovation, and an open, trusted relationship. Now, hold on to your questions. There's a Q&A session right after our final presentation, and I'm now handing over to Lucy Crabtree, Chief Financial Officer, for the next session.
Thank you very much, Aurélie. So I'm delighted to be here today to talk you through our financial strategy for the coming years, and how we will deliver growth and, of course, importantly, shareholder value with our long-term plan. So first, I'd like to start by looking back at our performance since our strategic refocus as a pure-play CDMO. We have delivered significant financial momentum over the last 3 years, with strong execution, commercial momentum, and disciplined cost control, translating into strong growth in revenues and profitability. On top line, we have delivered revenue growth of almost 90% since 2023, representing a 2-year CAGR of circa 37%. Significantly, we've also achieved positive operating EBITDA profitability for 2025, a milestone that demonstrates that we're not just growing at the top line, but we're becoming a profitable business, and have made rapid progress from our operating EBITDA position in 2023, when we made a £53 million operating EBITDA loss. This strong financial growth was delivered during a period of significant business transformation, and I don't think that should be underestimated. Not only did we deliver successfully an operating model reset within a volatile macro environment, we also expanded globally with the expansion into the EU with our Lyon and Strasbourg sites, and we, of course, more recently increased our capacity in the US with the acquisition of our Durham facility. So I'll not spend a lot of time talking about our prior performance, as I believe that the most important takeaway here is that we now have a strong foundation with the building blocks in place to continue growing further, capitalizing on the rapidly expanding cell and gene therapy market. So, as you will have heard from the presentations earlier today, we're incredibly excited about the potential of the cell and gene therapy market, and in particular OXB's competitive positioning as a viral vector CDMO. The viral vector CDMO market is expected to grow at a CAGR of around 18% through to 2030. We are highly confident that we will continue to outperform the market growth rate over the long term, and our ambition is a revenue CAGR of approximately 25% from 2025 through to 2030. Several key factors are driving our confidence in achieving this, which Sebastian spoke about earlier, and I'll touch on briefly here. Firstly, we're seeing a growing maturity in our client programs. In 2025, over 60% of our business came from late-stage projects. This is critical because late-stage work brings a high level of predictability and revenue visibility. It's more stable and provides better line of sight into our future performance. Secondly, our US facilities provide significant potential for revenue growth. In 2025, we more than doubled our US footprint with the acquisition of our Durham facility, giving us the capacity and capability to serve our clients' most advanced programs. We have the suites in place to service our expected growth, and with our flexible grey zone space in the US, we can quickly add additional suites to scale up as needed and capture the growth opportunity ahead of us. Thirdly, as Sebastian explained earlier, we also maintain exceptionally high client satisfaction. This isn't just about retaining existing business, though we do see high levels of repeat demand from current clients. It's also about our ability to win new business, with the majority of our recent wins coming from the US and Europe, as you have seen, which of course are the largest cell and gene therapy markets. Finally, we have great confidence in our platforms, which are supported by cutting-edge technologies and our 30-year track record, which Kiri and Nick spoke about earlier. So next I want to talk to you about our longer-term revenue vision. With a revenue CAGR of approximately 25% from 2025 through to 2030, our ambition is to achieve revenues of approximately 500 million in 2030. We don't see this as overly aspirational. As the business matures, we see this as a realistic outcome, supported by our track record, underlying market growth, and the capacity already in place. So on this slide, I'd like to give you an illustration of how this could potentially be achieved, and we have split this into multiple components. Firstly, process development revenues. In our illustration, we could assume this broadly tracks underlying market growth with CAGR of around 18%. We currently support around 40 early-stage projects and have a circa 30% proposal win rate, giving us confidence in securing new business. This first component would be estimated at contributing approximately £150 million of revenues annually by 2030. Next, pipeline maturation potentially contributes £150 million of revenue by 2030 as programs progress through the funnel and revenue per program increases. This illustration assumes around 40 early-stage programs, approximately 50% attrition, and standard batch volumes and pricing. Lastly, the primary swing factor is commercial scale-up. Just 2 to 3 mature commercial programs, each serving around 1,000 to 3,000 patients, can potentially generate roughly 200 million in annual revenue, representing the majority of the revenue build-up in this illustration. This is consistent with a bottom-up view. Even at moderate patient populations, GMP manufacturing economics scale quickly. Importantly, there are already multiple CAR-T programs either approved or in development for hematological oncology and autoimmune indications, which you've heard, relevant for the 3,000 to 5,000 patient range. With 9 OXB client programs already in late-stage development or under commercial contract, reaching 2 to 3 scaled programs is a credible near to mid-term outcome. Overall, this illustrative revenue build is deliberately conservative. It assumes standard pricing, modest batch volumes, and significant attrition across the early-stage pipeline, leaving potential upside from stronger conversion, improved pricing, or dose expansion. Critically, the existing manufacturing footprint already supports this growth, with no requirement for additional new build CapEx. As we move from our 2030 revenue ambition, I want to turn to how we expect to drive further margin expansion. Our long-term operating EBITDA margin target of approaching approximately 30% is consistent with other mature CDMOs, and here I'll talk to you about how we expect to get there. We've already made strong progress. In 2025, we delivered our first year of EBITDA profitability since the strategic turnaround or the strategic refocus, driven by disciplined cost control and improving operational execution. At a higher level, margin expansion is expected to be driven by a small number of dominant factors. Firstly, you have the mix shift to later stage and commercial work as the single biggest contributor, worth up to around 1,000 basis points, improving volumes, pricing and unit economics, and driving a corresponding reduction in cost of sales as a percentage of revenue. Next, installed capacity supports a utilization ramp of a similar magnitude, converting our largely fixed manufacturing cost base into operating leverage as activity scales across sites, particularly as new capacity ramps. On admin expenses, the corporate platform benefits from operational leverage. Costs grow materially slower than the top line, contributing up to around 700 basis points as revenue scales. For commercial costs, sales efficiency improves with higher productivity and a structurally lower cost per win as we move up the experience curve. This allows commercial investment to stay broadly stable as a percentage of revenues. We will continue to invest in innovation. You've heard how important that is to protect and build our market position. As Kiri spoke about earlier, innovation plays a pivotal role in our success and enables us to provide best-in-class services to our clients. We would expect for spend to stay broadly stable and gated to returns. This brings EBITDA margin approaching approximately 30% by around 2030 to 2031. In terms of cash generation, we would expect to convert a substantial proportion of EBITDA into free cash flow at maturity. Looking at our next slide, this shows that the pathway to this long-term margin guidance is not theoretical. It is a well-trodden CDMO maturity curve, and we're already on it. While cell and gene therapy is a newer modality, mature CDMOs across monoclonal antibodies, ADC, and CGT markets achieve margins above 30% at utilization maturity. This is a proven industry pattern, not a stretch target. Importantly, we are already seeing that operating leverage comes through. In 2025, incremental revenue flow-through exceeded around 30%, demonstrating strong margin progression even at our current utilization levels. Today, the business overall is still in the scaling phase, ramping utilization and just beginning to see that margin inflection. However, our UK site is obviously more advanced, providing clear validation of our model at scale, while other sites are earlier in the curve but positioned to accelerate as utilization increases. Looking ahead, the network is now scaling meaningfully. Durham comes online in the second half of 2026, driving US utilization, and France has roughly 3 times its installed capacity headroom at high incremental margin, enabling a true multi-site model. Under new commercial agreements, we have the ability to manufacture both in Durham and in the UK, which will facilitate further margin expansion by accelerating US capacity utilization. Finally, AAV and lentivirus tech transfer completion in France, alongside a shift towards commercial GMP mix and procurement scale, supports our 2027 expectation of at least 20% EBITDA margin, firmly on the path to approaching 30% in 2030 to 2031. Now moving on to CapEx. The delivery of our long-term expansion of the business is underpinned by a comprehensive capital allocation program aligned to strategic priorities and required returns. We have outlined a defined 70 to 75 million CapEx envelope across 2026 to 2028 as a disciplined investment cycle, with a circa 50 million CapEx across 2026 and 2027, followed by 20 to 25 million pounds steady-state CapEx in 2028. Investment is focused on scaling our US GMP manufacturing capabilities, including the commercial activation of our Durham site to meet client demand. We also intend to further strengthen our global CDMO network through flexible tech transfer across the UK, US, and Europe, enabling resilient, multi-factor delivery for programs from development through to commercial supply. Kiri and Nick mentioned earlier some of the ways that we're already using automation and AI in the business, and these will be further embedded across our operations to enhance capacity, planning, tech transfer efficiency, and process performance. And as mentioned earlier, we will continue to invest in innovation and platform productivity, including process intensification, analytics, and yield improvement to support improved cost of goods as the business scales. We will maintain our financial strength and flexibility by keeping our balance sheet capacity intact, ensuring the business is fully capitalized to execute our strategy while retaining the ability to support further strategic options. So, in summary, I believe we've set out a clear, achievable path to value creation. Our transformation into a pure-play CDMO is on the verge of being complete, and our commercial momentum is now driving growth in revenue and profitability. We're confident in achieving revenue growth that outpaces the broader viral vector CDMO market, underpinned by long-term client relationships and maturing programs. As mentioned earlier, we see a realistic path to revenues of circa 500 million in 2030. As site utilization increases over the coming years, we expect to achieve operating leverage, which coupled with continued cost discipline, will drive sustainable EBITDA margin expansion, approaching around 30% by 2030 or 2031, as we become a more mature CDMO. We have a customer-focused growth model that is structurally accretive to working capital and a path to robust cash conversion as the business matures. We will continue to execute our disciplined capital allocation path, prioritizing investments in growth while retaining an attractive EBITDA profile. So I'm incredibly excited about the future ahead as we move into our next phase of growth and continue to deliver as a leading global viral vector CDMO. So thank you. And with that, I think we're ready for Q&A. So I'll hand over to Aurélie.
Thank you, Lucy. And thank you to all of our speakers. It is definitely a very incredible time for OXB. We now our second Q&A, and I'll invite Frank, Lucy, and Sebastian. And we'll start with questions from the room and then move to the webcast question.
Hi, Charles Weston from RBC again. One for Arun first, if I can, please. So thank you for the color that you provided. Can you give us a sense of how much viral vector is as a proportion of your COGS, basically, at maturity when you've actually got product out there and patients being treated? And secondly, what would make you think about dual sourcing your viral vector supply? And my second question, please, for Sebastian, who's just handed the microphone over. Just on that pipeline chart that you provided, I think you said around about a third of your opportunities by number were phase 3 and commercial, which means presumably, given they're 5 to 10 times the revenue, a massive proportion of your pipeline by revenue is that. I mean, is my maths right?
Okay.
You want to start on?
Yeah. You want to?
No, go ahead.
Okay. You sure? Okay. So, hey guys. So a multi-part question. Let's see if I can get it all. First part is what portion of our COGS are driven by viral vector? We have not publicly disclosed our overall COGS and what exact portion it is. What we have communicated publicly previously is when you think about the early launches of CAR-T, a significant portion of the cost was in fact viral vector. Over the years, what has happened is that because of improvements in scale, in titer, in yield, we have seen the cost per patient, and Sebastian's metric is really the important one to think about that he referenced in either the first Q&A or in his presentation. The cost per patient is what we are focused on, and the cost per patient that is attributed to Vector over the years as CAR-T has matured has gone down precipitously. So it is not a significant driver in the overall cost of goods when we think about delivering a product to patients that is, that is in all aspects what we would want to make. So that cost has gone down significantly, which is really great. There are costs, and these would be modeled in sort of any way you think about it in terms of process development, process characterization that we have to work on together. So that's an independent aspect of it, but I just wanted to make sure we were clear there. Now I missed the second part. I missed the second question. What was the second?
It was at what scale or what would make you think about dual sourcing?
As in internalizing it?
Well, either.
I think it really goes back to the criteria that we talked about. So it's when we think about, I think, as a company, who we are and what we want to be, we're always thinking about risk-adjusted value. So whatever choice we make, we have to make sure that it provides us the highest value on a risk-adjusted basis that we have. The reliability and the experience that the Oxford team has makes that risk-adjusted value number go up because of the experience that's there, because of the reliability that they have and the experience with the agencies. So unless there was some significant change in that equation, it would be, I think, not a likely scenario. Now, look, no one ever knows what's going to happen, but I think that's at least hopefully helps you illustrate how we think about it. And then for the second question, I can hand over to Sebastian.
30% of the pipeline in number of opportunities is between phase 3 and commercial, indeed. So not in value, number of opportunities. The portfolio of late-stage activities is extremely diverse. You can find the process characterization, small-scale activities in Nick's lab. You can find repeat manufacturing. A process characterization process can be 5 million, but 10 batches are going to be, let's say, 20 million for the sake of the exercise. So the number of opportunities will not tell you much, except that indeed the value of a late-stage opportunity is way above the value of an early-stage opportunity. Why are we there compared to a few years ago? Because of the combination of today a number of programs that we were running in Oxford that have reached that stage, late-stage preparation for commercial activities or even building inventory for future commercial introduction, plus acquisition directly of new commercial programs, acquisition of the site in Durham, where we have attracted new clients. I talked about one where we have the kickoff this week on a new late-stage program. Indeed, it's a very significant portion of the pipeline. And if you look in number of projects, it's more or less 20% of the projects that we run today. One project can be 2 different opportunities because there may be one part of the project in UK, one part in US, which that has 2 different opportunities. And could be 2 different types of activities. Could be process characterization and routine manufacturing. Some clients ask to have 2 different proposals for these activities and not just one for multiple reasons. Indeed, it's a very big part of the pipeline. To my surprise, I must say, I did not expect that, and I believe it's part of Aurélie's team efforts that are paying today as the head of marketing. They did a very good job at rebranding Oxford Biomedica at OXB. That's making sure that we were recognized as a CDMO, as a global CDMO, and the perception from the market on OXB has really completely changed. So indeed, it's a big part of the pipeline today.
Yeah, thank you for taking my question. James Osborne from Stifel. First question, just around following up on those late-stage projects. We know obviously Arcellx have filed their BLA and probably going to have a PDUFA date at the end of the year. And then Caverna had some positive data and it looked like they might launch next year. Just wondering how you, as a CDMO, approach that. Are you manufacturing at risk, or how do you structure that from your side as they move into the launch process?
Manufacturing at risk is not really how we start the discussion with our clients. I mean, that wouldn't make a lot of sense to manufacture at risk, actually. You know what you need for the clinical studies. Based on the clinical data through phase 3, you know exactly how you're going to work on your market introduction. Based on your market introduction strategy, you know how many batches you need, so the risk is actually very limited. Except finding, and I don't see how that would happen, but except finding in the middle of phase 3 that finally you have a safety issue. I mean, that would be very late. Who knows? But there's not much risk. At this stage of the relationship with clients, they have a forecast that they communicate. It's binding because they need slots and they need material at a given date. We commit to a delivery date and we manufacture according to their forecast. So it's very scripted, very guided. They know where they go. They know exactly where they want us to go. We go in this direction.
Thank you. And then there's maybe one for Lucy on utilization, and obviously that's going to be a key driver to margins. Where are your peers at in peak utilization? I obviously see Samsung up there in the top right versus where you are today, if you can help us with that at all.
I think it's a slightly difficult question to answer because obviously the different CDMOs take different shapes. I mean, really that slide's just designed to show that indicatively, we can see what this path is. Now we've got our own journey to take as a pure play viral vector CDMO. And we know internally what we're striving for in terms of peak utilization. We've got very good utilization levels in Oxford, and obviously as Durham and our other facilities get to sort of peak capacity, those utilizations will look exactly as we are sort of guiding towards and working towards, and that's really what drives that margin expansion. And we have mapped this out. We have a long range plan where we're looking at this very, very carefully.
Thank you.
Hi guys, it's Sam England from Berenberg. Just one around CapEx plans. Can you talk about how much of the 500 million revenue ambition can be fulfilled with the existing capacity you've got, plus Durham and capacity coming online this year? Or are you assuming further, I think, called them grey site additions from 2027 onwards? And how should we think about CapEx needs beyond 2028 as the business continues to scale, both in sort of value terms, but also geographically where you might look to add sites?
Yeah, so look, we've mapped out our CapEx requirements, and we've got enough capacity to fulfil the 500 million ambition, albeit our CapEx requirements, particularly in sort of 28 and 29, incorporate expanding into the grey zones, right? So we don't need to break new ground or build new sites. That's the most important thing. And what we've steered to is obviously the circa 50 million over 26, 27, and then steady state thereafter. And that obviously captures how we need to sort of service and expand into those grey zones.
Thank you. It's Miles Dixon again from Peel Hunt. If I can just check on my understanding and the risk to the upside on the 500 million. Sebastian, I was surprised to see, I believe you said 10 to 150 million for commercial stage projects, and that you've currently got 3. Lucy, I thought you said 3 commercial stage projects in 2030. We might expect to see 200 million. And given that the retention of those late stage clients that you've got is 90%, might we expect to see 6 to 8 commercial clients by 2030?
So, Miles, great question. And I think, as you can see plastered all over that slide is illustrative, right? What we're trying to sort of give is, we have mapped out our 500 million revenue ambition. It's what, it's a target, it's an aspiration, and it's an ambition. And, we've looked at different ways of sort of, cutting the lemon or whatever the expression is to, we look bottoms up, we look top down. And, this really is designed to be illustrative. And as I said in my narrative, this is perfectly achievable in our view. Okay. Now, of course there's upside. This isn't a cap. It's a milestone that we're looking to achieve. So this isn't me steering and saying, look, we expect 2 to 3 commercial programs. This is saying there is a credible rationale for how we can get to that aspiration in 2030. But I don't know if my colleagues want to add anything.
If I look at how we built the long-range plan, which is giving the circa 500 million that was on the slide, a commercial program can reach 150 million, but then we're going to go back to one of the previous questions. What about fourth line of treatment, third line of treatment, and so on? So when you start and you introduce, you're going to treat the first year. I remember seeing a plan from one of our clients recently was planning the first year to treat a few tens of clients. To go to a few hundred, you needed 3 years. To go to a few thousand, you needed another 2 years. 2031 is in 5 years from now. Then it grows exponentially because the success you had in Europe or US, you're finding then in Korea, Japan, Middle East, and so on, and that's where the number of patients keeps increasing. What we should never underestimate is the time to move a program from fourth line to third and even second line, or file with other geographies. The timing that we discuss with our clients when we talk about manufacturing, we talk in quarters. When you look at the filing timeline, you talk in years. You can submit in Japan today, you won't get a response, or you won't get a program approved even next year, probably be the year after. Then you will start treating the first patient commercially, and to go to a few hundreds will take another 3 years. So that's the reason why, even if we see from what we're doing today, Arcelix was mentioned earlier, good data with Kyverna, good data with Cabaletta, and so on. We know that all these programs are going to be commercial quite soon, but we don't underestimate the market introduction timeline. So we see a bright future beyond 2030, but compared to biologics, cell and gene therapy is still a nascent market. And before we see the same number of patients treated with gene therapy treatment compared to biologics, there will be a few decades. Unfortunately.
Maybe we shorten it a little bit.
Yeah.
Hi, Christian Lenny with Stifel. Just trying to, you talked a lot about quite a lot of excitement around in vivo, CAR T particularly, and versus the ex vivo currently, sort of most of the products today. Does that change in terms of what sort of you have, your sort of expertise, your technology that you need to offer to be able to meet that interest? And obviously, we've seen a lot of acquisitions in this area, particularly as well. So is there anything that you need to adapt around that sort of in vivo versus ex vivo?
We don't need to adapt. The team has experience in vivo. Oxford Biomedica, as a company developing its own products in the past, was on in vivo therapies as well, so we don't need to adapt anything. In the discussion we have with the executive team, I'm probably the less excited by in vivo because of the timeline. If you look at the slides we presented today, 112 in vivo programs, 2,100 ex vivo programs. In vivo will come for sure, but we still have 20 times less in vivo programs than ex vivo. So I believe that the next 5 years are going to be still very intense with ex vivo. There were indeed acquisitions, as it could be expected. Many big pharma continue to not develop and on board through the acquisition of emerging biotech. Same trend as always, I'm tempted to say. 20 times less. So in 5 years from now, I think I will have very good reasons to be excited by the commercial pipeline in vivo. Today, we're engaged in multiple discussions with the in vivo companies, but as Kiri showed it, the number of phase 1 is, a handful, more or less. Yeah, it's the very beginning. So yeah, it's exciting, but with a 10 years horizon. Ex vivo is now.
Thank you. And then follow up on just a little bit unclear where you are with the non-viral offering opportunity. On the one hand, you said it's something you're working with today, but then I think Frank said that's not in the near term. You're the viral experts, right? So what are you offering today, and what might you need? Do you go and acquire something, or what's the strategy around non-viral? Just trying to really understand. I get the whole more holistic offering across all for everything you can do, what would you actually need to be able to be a credible non-viral partner?
That's a very good question. Let me start to make it very clear. I would say for the next 3 to 5 years, our focus will be on viral vectors. That's where our expertise is. It's where our differentiation against competitors is. That's what we will do. Now, we have also to think ahead of these 5 years. And then we look at what could be within the field of cell and gene therapy. Because without leaving this field, we might enter non-viral. Honestly, I believe the only way to do it would be by an acquisition. So far, I cannot see another way to do it. Yeah. Now, there is something in between which is called virus-like particles. Yeah. And we will enter this field in the next, or we are about to enter this field. And this will be the first step to enlarge a little bit, just not only doing viral vector, but I don't see non-viral before 5 to 10 years.
We're building the strategy to make sure that we're not missing the train of the non-viral vectors. You've seen it on one of my graphs. The space in which we play is about 50% of the gene therapy programs. We know that non-viral will continue to evolve, will continue to take some market share. So we will be ready on time. We also don't want to echo what Frank just said, to reinvent the wheel. There are people who do that fantastically well. It will be a good opportunity for us to grow by acquisition. Exactly what we've done with AAV. We acquired the CMC team of a company that had built an expertise and experience, a full platform on the AAV side, and that's the reason why today our AAV offering works. If we had started AAV from scratch, we would probably not have a platform yet today. Same for the non-viral space. So we're working with the strategy team on the potential acquisition strategy for the future. It's a long-term exercise. Yeah, indeed, if we were moving in 3 years from now, I would consider it's an early move already. We look into it. We're not close from pulling the trigger at all.
Julie Simmons, Panmure Liberum. You've made a really good case as to why Oxford Biomedica, due to the sort of longevity of the knowledge and the experience you've got. When you don't win clients, who do they go to and why?
I wish I know, but many times the clients do not want to communicate with which CDMO they're working. I'm actually glad that Arun and Cabaletta Bio overall accepted that we communicate on the relationship. But if you look at the numbers of logos that we display on our slides versus the number of clients, I mean, like 10%, 20% of our clients max say it. So we don't know, but we know the usual suspect. There are going to be Lonza, Catalent, and AGC Biologics. We usually lose for the very big ones. Because back to Charles' questions on the weight in the pipeline of the late stage opportunities, many companies now have understood that working with a CDMO just for phase 1 and then finding a reliable partner for phase 3 is not a smart move. Pick the right partner from the very beginning. And the right partner need to have the capacity, the capabilities, and the experience. And from the 160 CDMOs in cell and gene therapy worldwide, if you select only those who have the commercial experience and the commercial track record, then you're going down to 10-ish, not more than that. So we know that usually we lose to the big ones. Yeah, the 3 names I would mention are the one I mentioned already. But many times we don't know.
And in terms of sort of the Durham site, has that generated more interest globally from new partners, or is it mostly those who are wanting US active?
US. Clearly. The European clients are still very interested by Oxford, Lyon, and Strasbourg. So the demand is primarily coming from US.
I would say we have time for 1 or 2 questions from the room.
Hi, it's Ken Slutsky from Deutsche. I don't want to take away the theme of the long term, but if I could just quickly sneak one in on near term, you're probably going to shoot me down, but any commentary on sort of current trading, given we sort of 5 months into the year? Is it still the H1 sort of loss into the H2 swing for the full year this year?
Yeah, sure. So, I mean, look, the Capital Markets Day, we're here to sort of talk about the long term. We obviously spoke about current guidance at the full year result. And I mean, it remains the same. We're not changing any of our guidance. And yes, it will be second half weighted.
Sophia, there was a last question. Sorry, Aurélie. But I saw him raising the hand many times and he never comes.
Thanks a lot. So the slide showed significant late-stage opportunities in your pipeline. Can we assume, therefore, that there are many of your projects, your early-stage programs that are in your competitors' late-stage pipeline, and do you have visibility over that?
The late-stage opportunities are coming either from programs that have demonstrated safety and efficacy in phase 1. It's one of the beauties of gene therapy. You can see the efficacy as early as phase 1, but also from the people coming to us. I mean, as I just said, we had a CDMO for phase 1. They can do phase 3 and so on. So that's really a mix. We see that I don't believe we became more attractive. We just became more visible. And for that reason, we see more people coming to us saying, why don't you take that? On top, and that's the third layer, which we should not underestimate. People who say we need a lot of capacity and we're working with a CDMO, they have limited capacity. Would you be a second supplier? So it's not a question of dual sourcing in the sense of business mitigation. It's really capacity play. And we've built capacity at the same time as we built expertise. So that's really a mix of these three that we see today.
So as a early stage program is showing promising data, presumably you are in deep discussions with those clients on getting them into your late stage program.
Well, actually, many of them from the very beginning, and it's probably because of our approach as well. We tend to sign with our clients master services agreements that cover all the phases so that we don't need to renegotiate afterwards. I mean, in full transparency, I have a call with one of our very new clients. We just started a feasibility study a couple of weeks ago, 3 weeks ago now. And they want to incorporate into the master services agreement the prices at commercial scale. Ultra rare disease, fast track designation. They want to know what commercial is going to be like. So meaning that commercial is part of the early stage contract as well. Not unusual. Not the first time I see it. You wouldn't see that with small molecules or biologic. You see it in gene therapy. And for that reason, we know that we can bet on late stage coming from early stage. Yeah.
Thanks. That's very helpful. If I could slip in one more. Clearly.
Ask the boss.
Boss?
Please.
Yeah, it's a quick one, I think. Clearly, the acquisitions of Lyon and Durham have been transformational to your positioning. Are there other stranded assets out there that competitors could acquire that could therefore transform their businesses?
Very good question. You've seen recently a lot of companies divesting their CGT business. They had the capacity, they did not have the expertise. Because we had the expertise and the demand, we acquire capacity. It's a very different thought process. Many of our competitors today, including some names I mentioned earlier, are struggling because of the turnover of the employees. I didn't say it. Arun said it. The tenure at OXB is exceptional. Thierry, 25 years. You're soon approaching 12 years with Nick. We built an expertise that attracted a number of companies to us. For that reason, we need more capacity. We acquired that capacity. That was Lyon. When we acquired Durham for 2 reasons. We needed commercial capacity in the US, and the site was not coming as an empty shell because we had proposals for many empty shells, and we systematically said, no, we're not going to bid. We acquired Durham because it was a building coming with experience and a very experienced team, ex-Bluebird Bio employees who had stayed with Resilience, who stayed with Oxford Biomedica. Out of all the employees who were on site, we lost 1 at the time of closing. So it was really about making sure we were keeping the expertise. It was the same with Lyon and Strasbourg. Strasbourg, the site was GMP for viral vectors in 1995, 31 years ago. Empty shells, not interested. Effective teams, absolutely. There are not so many effective teams on the market right now.
More questions? Yeah. Okay. We'll move to our webcast audience then. Question for you, Sébastien, from Harry Gillis from Berenberg. Are later stage programs with higher volume more profitable on a per patient per dose basis due to economy of scale, as well as having larger contract size? And does OXP's manufacturing capability per unit of manufacturing footprint increase if there is a mixed shift to fewer, larger, late-stage programs?
More profitable. Second one was?
More profitable on a per patient dose basis due to economy of scale, as well as having larger.
Yeah, and bigger size of the contract, right?
Yeah.
Not always to the second part. It's not always a bigger contract. I have in mind one of our clients, ultra rare disease. The need is one batch every year, maximum at commercial scale, maybe one batch every 2 years. Some very early stage program need 2-3 batches. So you cannot always say because it's late stage, it's going to be a bigger contract. Not true. Particularly in gene therapy, not true. If you look average to average, indeed, the average late stage is much bigger than the average early stage. That's very clear. Economy of scale is a very good question, and the answer is absolutely not straightforward. If you make the decision, Cabaletta made that decision actually very early in the process to look at efficiency, automation in the cell manufacturing process, for example. Then you're going to look at the cost of goods from the very beginning of your process development, exactly what you did. Then you're going to benefit economies of scale. At the beginning, you know you're going to start at 200 liters, you're going to get X doses, you're going to automate the cell manufacturing process. But it needs to be by design in your process development and manufacturing plans. If you don't plan for that, you won't see any economy of scale. It's always very surprising, but I still see many people, and let's say 90% of the discussions we have, when we say if you were scaling up from 200 liters to 1,000 liters, you would mathematically divide the cost per dose by a factor of 2 to 3, and people say, I'm going to stay at 200 liters. So to benefit from the economy of scales, you need to proactively make that decision. It needs to be part of your development plan. And that's not always the case. Is that more profitable? By definition, running routinely manufacturing is more profitable than working on a process development project. Let's go back to a couple of numbers. If you take 100 programs at process development stage leading into a phase 1, how many will fail in phase 1? 9 out of 10. So indeed, the level of risk being very high, these programs are by design less profitable than routine manufacturing, where the success rate in manufacturing is 95+%.
And another question from Chris Richardson from Jefferies. The Stellaris cell shuttle take makes a strong case for enabling scale in larger indications from Cabaletta's perspective. Is automation now a prerequisite for pursuing those high prevalence indications? And for OXP, how do such developments in cell manufacture change how you operate or adapt on the viral vector side? As a follow-up, to follow up on the earlier cost question, is it such platform automation that drives the margin expansion, or are bespoke projects required to drive this expansion?
Is automation a prerequisite? It's not, but it's a wise decision to go for an automated process. It's not only, we're not only talking about economics, we're talking about probability of success or failure rate, the other way around. with automation comes a much higher success rate. So it's a wise decision to shoot for automation from the beginning. But I'm not going to put words in your mouth, Aaron, but again, needs to be part of your very early decisions, right?
Yeah. Just to talk quickly about it, it's hard to say what other companies do or don't think. I think for Cabaletta, we made the decision early on when we saw the scale of the opportunity that in order to meet the need for patients, we needed to invest heavily in automation. Automation is not the only thing we're investing in, and our initial product, our initial programs are starting with a traditional, we'll call it manufacturing process, so that's not fully automated. It's, again, hard to comment on what others would say, but for us, making that decision and that investment early was critical because we saw the scale of the opportunity and wanted to be able to deliver to that opportunity.
Is that changing anything for OXB? The answer is yes and no. In theory, it's not changing much. We're still providing the viral vector to our clients who then transfer to Celarius, who's going to take care of the cell manufacturing process. Practically speaking, I believe there is an impact. As I said, the success rate is going to be higher. So if the success rate is higher, I personally believe that then the treatments are going to be more affordable, which means that this treatment is going to be successful. If a treatment is successful, OXB is successful.
Thank you. Thank you all for your questions. Thank you to the speakers. And I'll hand over to Frank to summarize and close the session.
Yeah, thank you. Thank you so much. I hope it was a great session for all of you, which is now coming to an end. But before I let you go, I have a last one thing, because I have one slide which for me is the most important slide. It's a slide which motivates me. I wouldn't say every day because I'm motivated by definition, but which I show to a lot of our people, of our newcomers, every time they join the company, is why we are so confident, why I am so confident and so excited about the future of the company. But not only me, the team, the executive team, key shareholders are excited about us, and the board is excited. And this slide is a nice way to summarize everything. It's why we are so excited. And it's very simple, but it summarizes everything. High unmet medical need. There are so many patients still suffering from rare diseases, for oncology, from cancer and things like that. There's a big, big need for such kind of therapies. And as it was said a few times today, and sometimes it's one shot and it's done. You are cured. Yeah, this is the vision which is accompanying us. We have skilled and experienced people. Believe me, I hear a lot of clients. Hopefully you say the same, I would. Yeah, you have the best people. You just have to think that all the CDMOs have very similar bioreactors, very similar suites. Where is the differentiation coming from? It's coming from the people. And here we can build on 30 years of experience. 30 years is a lot just doing one thing. It started with lentiviral vectors. We added AAV. Through France, we became also more agnostic into others. We did, during the pandemic, the adenovirus for AstraZeneca vaccines. Yeah, we build no other CDMO can build that within a short time period. It's not possible. I think we heard a lot about, thank you, Kiri, the innovation part, which will continue to guide us over the next years. We are totally vector agnostic. We are geography agnostic. We can serve our clients from everywhere, from our sites, with the same level of quality and service. We have now adapted our processes. The transformation was a difficult one, because when you are a development company and you want to transform to CDMO, it sounds very simple. At the end, we do the same. No, but we do it totally differently. And so we need different skills. We need different ways of working. We need different processes. We have adapted all this now. Global footprint, we are in the main hubs of the world for cell and gene therapies. And maybe one day we will go to Asia. Sébastien, it's in your direction. Yeah. Client satisfaction. Thank you, Arun. It was so nice to listen to you Yeah, but this client satisfaction is very high and bringing a lot of clients back. And we have, thank you, Lucy, a clear path to profitability. So tell me, if this is not a compelling case, I don't know what a compelling case is. We are at the right time, at the right market, with the right service. So thank you so much. It was a pleasure to host you today here in London, and thank you for those on the web to join us. So we have the possibility to make a virtual tour outside. Some of you have done it already. I've seen that. So if you want to go virtually in a tour, we can offer this already. Yeah, we have people experience outside. But there's another way to do a virtual tour is to come and have some drinks with us. Yeah. So thank you so much for being with us, and thank you for your interest in the company. It was a pleasure to have you with us today. Take care.